はい、皆さん、こんにちは。今日はですね、このシンポジウムにご参加いただきまして誠にありがとうございます。実はですね、私は今日はとっても嬉しいんです。なぜならば、久々にですね、具体的には3年半ぶりにですね、この日本橋において UC サンディエゴ校の皆様と、こういうリアルおよびオンラインでの、シンポジウムを開くことができることになりました。実はですね、UC サンディエゴ校様とはですね、Link-J 設立より前からですね、大変、一緒にいろいろとですね、アドバイスもいただいて一緒にやらせていただいております。特にですね、今日ご登壇いただきますですね、ジェイコブスクールオブエンジニアリングの、ピサノ学部長、そしてですね、全学の、リサーチアフェアーズ、のシニアディレクターをなさっている和賀さんにはですね、大変お世話になって、お助けをいただいております。この場を借りてですね、御礼を申し上げます。本当にありがとうございます、いつも。そしてですね、Link-J なんですが、ちょっとお待ちください。今、設立以前からというふうに申し上げましたが、設立して今ちょうど7年と3ヶ月、を迎えております。現在ですね、会員数、これ6月1日で661になってますが、今日現在で674、の会員数になっております。多くの方にですね、会員になっていただいておりまして、Link-J のメンバーの方、および Link-J 自身がですね、昨年1年間で834回、こういったライフサイエンス関係のですね、シンポジウム、イベント、アクセラレーションプログラム、インキュベーションプログラム等々をやってまいりました。設立以来ですね、グローバル展開には先ほど申し上げましたように、かなり力を入れておりまして、その中でもですね、UC サンディエゴ校さんとのコラボレーションがダントツでございまして、ウェブを見る、調べてこう計算するだけでもですね、過去、24回ですね、Link-J のウェブでイベントが、やったというのが載っているというところになります。さらにはですね、昨年の9月なんですけれども、これサンディエゴにあるですね、UC サンディエゴ校のキャンパスの中にですね、フランクリン・アントニオ・ホールという新しいですね、工学部を中心に、医学だけではなく、他のさまざまな分野とのですね、学際間を超えるコラボレーションの素晴らしいセンターが新設されました。そちらの一角にですね、Link-J の拠点もですね、置かせていただいているというところで、ございます。そしてですね、ホットなニュースがもう一つございます。昨年の1月ですね、岸田首相がですね、今年はスタートアップ元年だというふうにおっしゃいました。それ以降ですね、日本政府いろいろな具体的な施策、どんどん打ってきておりまして、その中の一つがですね、起業家を1000人海外に送って武者修行すると。いうのがございます。それがいよいよですね、具体化して動き出します。これはですね、いろいろな地域、いろいろな領域ですね、それぞれに分かれて全世界にそういった起業家の人たちを送り込むというプログラムなんですが、その中でもですね、バイオインダストリー、創薬、再生医療、医療機器を含め、デジタルヘルスも含めてですね、は、ここサンディエゴがですね、中心になって人を送ろうというプログラムで、いくつかのモジュールに分かれている中で、ビヨンドジャパンという、モジュールがございます。ちょっと右上の方に書いてあるんですが、渡航費参加者ですね、海外の往復無料、宿泊費無料、プログラム費、メンタリング費も無料という非常にですね、参加者にとってはありがたい内容になっておりまして、期間もですね、2週間から8週間まで、参加者の方々によって応じて決められるというものになっています。ところがですね、応募の締め切りがすぐでございまして、来月の10日、7月10日になっております。で、今申し上げましたように、この、ビヨンドジャパンはですね、ロサンゼルス、サンディエゴ、テキサス・オースティン、この3都市がこのモジュールには含まれております。ロサンゼルスはエンターテインメントなどを中心に、そしてオースティンの方は航空宇宙を中心に、そしてサンディエゴは今申し上げたバイオ、こちらを中心に、で、実際のアクセラレーターですね、ちょっと下の方に書いてあるんですけれども、UC サンディエゴ校、その先ほどの、ホールの中の1階にですね、インスティテュート・フォー・グローバル・アントレプレナーという、素晴らしい組織があります。いろんな大学、日本の大学の方がもうすでに派遣されてですね、育成プログラムを受けられています。この、インスティテュート・フォー・グローバル・アントレプレナーはですね、先ほどお話もご紹介申し上げたピサノ学部長の工学部とですね、レイディースクールというビジネススクールですね、UC サンディエゴ校、その、工学部とビジネススクールがコラボレーションをしてやっている素晴らしい、高付加価値、バリューの高い、組織となっております。ぜひですね、皆さん、こちら、 今日ご参加のご視聴のご自身、皆さんでも結構ですし、周りにいる方でですね、なんかこういうの言ったらいいんじゃないのって興味がありそうな方、言ったらいいと思われる方にお声掛けをいただければ幸いです。先ほど7月10日からと申し上げたんですが、もう一つございまして、締め切りがですね、7月21日。12日間しかないという、まあちょっと役所にありがちかもしれないんですけれども、そんなプログラムになっております。ぜひですね、いいプログラムですので、ご参加、またお声掛けをいただければと思います。私からはですね、これで以上とさせていただきます。では続いてですね、今、何度かご紹介申し上げました、ピサの学部長からですね、オープニングリマークスおよびですね、その後の第一セッションのモデレーションの方をやっていただきます。ピサさんよろしくお願い申し上げます。
Well, greetings and welcome. I'm so happy to be here. It's nice that we can travel again, and it's so great to be working with people in person in Japan. Sayama-san, thank you very, very much. I am Al Pisano. I am the Dean of the Jacob School of Engineering, and I think we have been strong strategic partners, you and I, for a little over 7 years, as you mentioned. Time flies. So today we're all gathered here to talk about a very important topic. This topic is important both to Japan and to the United States. We are going to be discussing recent advances in big data analytics and synthetic biology, looking specifically at platform technology for biomanufacturing for the future. This is a very big, important topic. And let's pause and think just for one moment. What is a platform technology? We will discuss platform technology, but let me ask, what is a platform technology? So a platform technology is a set of related technologies that work together that can be reoriented from one purpose to another. And if your technology platform has 10 separate technologies in it, you must reorient each of the 10 from one target to another. So what are examples of platform technologies? Well, messenger RNA, mRNA, is a set of technologies that was pivoted, right, from cancer to COVID. But there are many others. The semiconductor industry, microelectronics can be pivoted to wireless or to communications. Wireless communications themselves is a set of technologies that can be pivoted to new uses. And even the cloud, which is a combination of computation, storage, and optical fiber communication, even the cloud is a platform technology that can be put to multiple uses. So why are platform technologies so important? And I feel that they are important because platform technologies create great national wealth 2 times over. They create great national wealth because of the technology itself. Wireless communications, a big business. Semiconductors, a big business. mRNA, a big business. Biomanufacturing, another big business that's growing ever bigger. But it generates a second increment of wealth because the platform technology creates wealth not just in itself, but in the application space. So COVID vaccine manufacturing is another wealth generator, for example. So today, we will hear from a number of world experts. They will look at biomanufacturing from a number of different viewpoints. And as you hear from our experts today, I am going to encourage all of you, not just the people in the room, but the people attending remotely, please think upon this. Innovation is a race that is won by those with speed and accuracy. You need two skills to win an innovation race. So, I would like everyone to start thinking about a question. And I'm going to ask this question in session 3 of the panel. I'm going to ask, what new infrastructure do we need so that we can build a new research and innovation platform technology? This technology is going to help our two countries join forces and enable us to go faster and better together. So, what is that new research platform technology that biomanufacturing needs so that the United States and Japan can be partners and go faster and better together? That is the big question that perhaps we can start answering. But before we think of that question, we have to learn a little bit more about biomanufacturing. So our experts are here to teach us. And the first speaker on the list is going to be Michael Burkhardt. So Michael, would you come on up and make your presentation? And please remember to make just a 1-minute introduction of yourself before you begin your talk. Thank you very much.
Thanks, Al. It's really a pleasure to be joining you today, both here in the room and online. So my name is Mike Burkhardt. I'm a professor of chemistry and biochemistry at UC San Diego. I'm the chair of the department. And I also am part of the materials engineering program in Dean Pisano's school. And I've been a professor at UCSD for 20 years. My research involves natural product biosynthesis, and we use chemical biology as a tool to understand and develop these pathways. And I'll be telling you about that today. I'm also the associate director of the California Center for Algae Biotechnology, where we're using the algae as a source for making renewable fuels and materials for our renewable future. What I'm going to be talking about today, however, is really a focus on secondary metabolism. And really, I'm going to try to discuss 20 years of research in about 15 minutes. So wish me luck. But what I'll tell you is how we have used the tools of chemical biology, organic chemistry, being applied to questions of biology. And we want to use that then to get us to where we can engineer pathways and make molecules that are important to all of us. Just a quick reminder, this is a graph that shows small molecule development between 1981 and 2019. So these are drugs that have made it through clinical trials and into the market. And what you'll see is everything except this bottom yellow slice represents molecules that either are natural products, are derived from natural products, or are based upon natural products. And natural products have been a huge source of pharmaceuticals for centuries, and they will continue to be an area of growth and interest. My lab is focused on the types of molecules that you see on this slide. So we're interested in polyketide molecules. For example, this molecule erythromycin or tetracycline, two important antibiotics. We're interested in non-ribosomal peptides. Probably the most well-known is penicillin or vancomycin. And we're interested in fatty acid biosynthesis. And these might not look similar to you, but it turns out that fatty acid biosynthesis is the evolutionary progenitor to all of these natural products. And nature evolved fatty acid biosynthesis to make the great diversity of molecules that you see on this slide. And it's an enormous class of natural products. These molecules are made by pathways that we call modular synthases. And what's really interesting about these is that these are linear pathways that make these molecules one monomer at a time and build them up in an assembly line. And this has been really exciting to me and into a whole field of people interested in natural products, because we have the potential to understand how these molecules are made and potentially the ability to engineer them. So this is a little illustration, a little cartoon of how these assembly lines work, where you have individual monomers. So in this case, we're looking at amino acid biosynthesis, and these amino acids come in and are activated by this adenylation domain marked A, and they get attached to a very small domain called carrier protein domain marked T for thiolation. And in between these two, you have a condensation domain C that links them together through covalent bonds in order to make a dipeptide. And now this dipeptide is transferred to the next domain, which forms a covalent linkage. And you can have some really complex and interesting chemistry, for example, to make a heterocycle, like you see in this case, in this thiazole. But at the end of the day, these are really exciting assembly line molecular factories. And if we can harness these by engineering them, we have the potential to make almost any type of molecule that you can imagine with different activities. The problem, however, has been that the synthetic biology of these systems is extremely complex. These are very, very large proteins, and they've been hindered by technical challenges. And actually, most groups kind of abandoned the potential of engineering maybe 15 years ago because it looked like it was too difficult. And groups like mine decided to really focus on the fundamentals of these pathways to understand what we can learn about them and eventually get to the engineering part. And I hope that I'll be able to prove that to you today. So what we decided to do is go back to this evolutionary progenitor. This is the most fundamental biosynthetic pathway, and that is fatty acid biosynthesis. Fatty acids are made by all organisms with similar, all of them have identical reactions occurring to make fatty acids. This is an iterative pathway that iterates over and over again to build fatty acids 2 carbons at a time. A hallmark of all of these pathways is they have a carrier protein, in this case, acyl carrier protein, that lies at the center, and it's surrounded by enzymes that act one at a time to elongate these carbons. This is a picture of what it looks like from the carrier protein itself. And here you see this is an iterative pathway of 4 enzymes, each one acting one at a time until it adds 2 carbons, and then it starts this process over again. One of the things that we found particularly interesting about this is that unlike other metabolic pathways, the intermediates are all covalently attached to the carrier protein. And in order for any of these enzymes, or what we call partner proteins, in order for them to catalyze reactions, they have to form a protein-protein interaction. And that specificity is where the timing and organization is coming from. And so we set out to try to understand that specificity. We did that by designing small molecule tools. And as a pathway into this process, every carrier protein in all of these pathways has to have a modification to them. This is a post-translational modification that adds a portion of coenzyme A to a conserved serine residue. And we saw this as an opportunity to get into this pathway through this transformation. And what we do is we synthetically make these precursors that are attached to pantethine, which is a part of coenzyme A. And then using enzymes in a chemoenzymatic process, we can attach these to the carrier protein. And now using this synthetic tag, we can start to study how this carrier protein is interacting with the enzymes within this pathway. So I'll be really showing you only a small portion of the research that we've done, and it's really focused on structural biology. What we do is we synthesize small molecules that are designed to specifically react with each enzyme in a covalent manner, such that the enzyme active site will form a covalent bond and trap the interaction of the carrier protein with the enzyme. And then we perform structural biology studies on that. And in this case, I'll be showing you some of our crystallography that we've done to try to elucidate these complexes. We've made many, many different probes. So again, this is our application of synthetic chemistry to design and synthesize these probes. And each one is designed for a specific enzyme system so that we can capture those interactions. So again, just focusing now on fatty acid biosynthesis, these enzymes that are highlighted in yellow are all ones that to date, we have captured in those interactions, and we've collected structural studies on them. These in white, we're still working on, but we're actually close to solving those. And they're all shown here. What I like to highlight is that all of these enzymes, so these are all coming from E. coli fatty acid biosynthesis, a very well understood system. And in fact, the enzymes themselves have been crystallized and studied decades ago. But before our work, no one had ever seen them with the carrier proteins attached to them. And these are their natural substrates. So the other thing that I'll point out is many of these are homodimeric proteins. So these are going to be dimers of each protein in which each monomer is covalently attached to a separate carrier protein. This one on the right is only a monomer. So just to break these down a little bit, why do I have two enzymes here and two enzymes here? Well, what's really interesting about fatty acid biosynthesis in E. coli is that all organisms have to have double bonds or unsaturations in their fatty acids. We need that so that the membranes can retain fluidity and pass molecules across the membranes. The way E. coli does that is it's got two of these dehydratase enzymes. One of them, FABZ, catalyzes the normal pathway, which goes from this alpha beta unsaturation, which then gets reduced in the next step. However, the second enzyme, FAB-A, has the ability to isomerize this intermediate into this cis beta gamma double bond. And even though we knew this information before, we never understood the chemical specificity. We never understood the mechanism. And we were able to figure that out by designing tools for each of these two enzymes. So the one at the top was designed for the enzyme Fab-A. The one at the bottom was designed for the enzyme Fab-Z. And we're able to capture both of these structures. Now, importantly, now that we have these structures, we're able to use the tools of computation to understand how the small molecule intermediates fit into the active site. By doing that, we can now start to understand the mechanism, substrate specificity, how this pathway progresses, and it gives us an opportunity to design these pathways for new substrates in the future. And so we're really using the tools of structural biology and computation to help us understand this very fundamental enzyme in the pathway. Also, in order to have this cis double bond, we need an opportunity to immortalize this double bond in the fatty acid. And that's why E. coli has two ketosynthase enzymes. One of them catalyzes the normal transformation, which is just an elongation from the saturated fatty acid. And the other is an elongation of this cis double bond that was formed by Fab-A. And so we were able to capture both of these two structures by designing selective probes for each one. In this process, we were able to learn something completely new about these enzymes. And I should point out, these are very complex enzymes that catalyze what's known as a ping pong mechanism. And this is what allows the fatty acid to grow in a very uniform direction, where the substrate first comes on, transfers onto the enzyme itself. The carrier protein falls off and brings in a new monomer so that it can further elongate. And our structural studies elucidated this brand new mechanism that had never been seen before because no one had gotten a structure with the carrier protein attached. And what we see is these two loops that move out of the way in order for the elongated fatty acid to come into the active site. And we see this as a gating mechanism for this pathway. And as I said, this had never been seen before. In fact, it turns out, though, that this mechanism is conserved across all enzymes in these pathways, all fatty acid synthases. It's also conserved among secondary metabolic processes that make polyketides. So we've got a fundamental understanding of the mechanism, and then we now have the ability to use this to our advantage. So these are all the different proteins in E. coli that are known to interact with this tiny little acyl carrier protein. And one could probably spend an entire career just studying these. But I want to point out how we've developed the tools that allow that to happen. Now, what I want to switch gears to is looking at secondary metabolism. So we were looking at fatty acids. Now let's look at polyketides. These are the molecules that we use for drugs. And here are just a few small molecule drugs, antibiotics, and an anti-cancer drug that are made through a type 2 polyketide synthase. And again, these are evolutionarily related, very similar to each other. This is a project that we have done with a Japanese collaborator, so Yasuo Onishi at the University of Tokyo. We worked together where a student came to my lab and conducted this research with me, and we were able to use these same tools to study the biosynthesis of this antibiotic known as ishigamide. And this probe allowed us to collect this crystal structure and look at the interface between the carrier protein and this KS-CLF enzyme. What's unique about this structure is that there's a high degree of specificity. So there are individual residues that form salt bridges to form these very complex protein-protein interaction. And nature uses this to keep these pathways independent from each other so that the polyketide synthase doesn't interfere with the fatty acid synthase. But we have to understand this information if we ever want to engineer or manipulate these pathways. The last example I want to talk about is from a non-ribosomal peptide synthetase, and this is a pathway that makes this antibiotic pioglutiorin, and this makes this pyrrole intermediate. So this is an aromatic intermediate. It comes from a simple amino acid, L-proline, and it gets loaded onto the carrier protein by this adenylation domain, PLTF. So we first began by just collecting the structure, and here we used this synthetic probe in order to collect that cross-link structure. But this really then allowed us to understand what's going on with those protein-protein interactions. And now we turn to the engineering part. So we actually had a very ambitious goal, and that was, can we now swap out the carrier protein from one pathway with that for another? And we chose the acyl carrier protein from fatty acid biosynthesis in E. coli. This is a carrier protein that should not have any activity with this enzyme from pioglutiorin. So our goal was to see, can we mutate the surface of the PLTF enzyme so that it now will load proline onto the E. coli fatty acid ACP. And again, we turn to our computational tools. So here we use a program that's called Rosetta. And what we do is in silico, we bind these proteins together, and then we have the computer predict mutations to be made on the surface of this PLTF protein. And that iterates 1,000 times, computationally selecting the best residues. Once these have been predicted by the computer, we then take it into the lab and we make those mutations in the protein, and we test to see how well it works. And then we iterate this process as many times as are needed to get the interactions that we want. And so in this process, we iterated this through 3 rounds. On the left was by using a certain scoring mode for the 2 proteins that focused more on hydrophobic interactions. And on the right, this score focused more on charge. And we see that we are able to increase activity. In this case, we got over 2,000-fold activity from this 3 mutations on the PLTF protein. And we're able to create new reactivity that never existed before. So this was really just to show you that this is possible. And now that we have these tools available, and we can really now use the power of computation, of AI, and the automation capabilities of AI, along with big data, we can now start to make this into a much more robust and rapid process. And this will allow us now to start engineering these very complex and elegant molecules for our purposes. And so, as I said kind of in the beginning, while this engineering had kind of been abandoned, we now have the tools that we can come back to it and start using computation along with the structures that we've collected to create new interactions and make new molecules. And so we're hoping then with the tools of chemical biology and structural biology, we're able to do in vitro evolution of these pathways and be able to apply them to synthetic biology. So what I want to do is bring this back around to the topic at hand, which is how can we leverage this with big data and our ability to work together, because these questions involve more than a single laboratory. We really need to be doing this together. And I think that's a real opportunity for this group to be thinking about these types of collaborative research efforts. I'll finish by just mentioning that I'm also the Associate Director of the California Center for Algae Biotechnology. Here we use green algae as a host organism for making biomass that we're converting into renewable products. We started back in 2008 focusing on biofuels, but more recently we've moved into renewable materials. And importantly, we focus on making bioplastics that are completely biodegradable. So we're looking for materials that can be used as drop-in solutions for everyday products, but that at the end of their useful life, they can rapidly biodegrade in a compost or even in the ocean. And so that's one of our visions. In fact, I'm wearing a pair of shoes that we've designed from the materials in the lab. I'd be happy to talk to you about that during drinks. So with that, I'd like to thank the people that actually did the work. Their names are listed here in blue. I also want to point out our collaborators. In this case, Joe Noel is a crystallographer at Salk Institute. Andy McCammon is in UCSD in my department, who's the computational expert, and then Yasuo Nishi at University of Tokyo. And finally, thank you all for your attention. I'd be happy to address any questions now.
Okay, so colleagues, both at home and in the audience here, are there any questions? We have 5 minutes for questions. So while you're thinking of a question, I'll ask one, and then maybe you'll think of one. So Michael, I was very impressed with your ability to mutate one of the parts of the manufacturing cycle, as it were. So I want to ask a question about that mutation. Occasionally I've heard in microbiology, when you put in place a mutation of one kind or another, sometimes the organism unmutates. It has a certain stubbornness.
Yes.
Are you seeing this mutation as a permanent mutation, or does it occasionally revert? Can you comment on that?
Absolutely. So first of all, we try very hard to just mutate those enzymes that aren't going to affect other parts of the metabolism, and that can be a challenge. But that also points to the tools of synthetic biology. It's actually very easy. Oh, I shouldn't say that. It's not terribly difficult to get enzymes to work inside a host organism and even make small amounts of a desired product. The real challenge is streamlining the metabolic process so that you have improved metabolic flux, right? You feed the organism glucose and you want your product to come out at the other end. That can be a very long and challenging process. And it gets me back to this idea that we need multiple people with skills in these different areas to solve those types of problems, right? So Bernard Paulson, who's going to give a talk in a little while, this is what he's an expert at.
We have a question? Excellent.
So yeah, thank you very much for a nice talk. So I was kind of surprised to see like only the 3 mutations improve like your efficiency by 1,000-fold, more than that.
2,000-fold.
2,000-fold.
Yes.
So how long is the protein by itself? How many amino acids?
How big is it?
Yeah.
So this enzyme is about, I think it's about 30 kilodaltons.
Right, not so big, right?
So I didn't have room in the slide. I have got I've got other slides. But importantly, this is really changing the charge of the surface. So the acyl carrier protein is highly negatively charged on the surface. And what these mutations did, if I can go, you can see these, we've got these positively charged residues on the surface.
Yeah.
And so
Wow.
this is not something that
You expected.
It's not that we didn't expect it. We couldn't have predicted where they needed to be, right? And that's why this iterative computational method is so powerful.
Yeah. That's right. Yeah, thank you very much. Great.
Okay, perhaps a question from the remote audience? Who is monitoring for the questions? Do we have any questions online? Okay. All right, one more request from the audience in the room. Any other questions? So I'll ask one more question because we should get as much information from you as possible. So when you have succeeded in making this machinery work, what kind of things can you think about making via biomanufacturing? In other words, where does this result take you in terms of manufacturing?
Sure. Well, let me give one example that actually comes here from Japan. So another researcher at the University of Tokyo named Hiroki Suga has a company called PeptiDream, and he's got this fabulous technology for designing peptide molecules, and they have a pipeline of maybe 30 potential drugs, all of them made as cyclic peptides. Now, the problem is to make a cyclic peptide synthetically is very expensive and very challenging. Microorganisms are great at making cyclic peptides that we use as antibiotics. And so if you could make them fermentatively, you're going to have a resource to actually make these and make them at scale very easily. So you could imagine that you could design some of these cyclic peptides that are being developed at PeptiDream. I've spoken with Professor Suga about this in the past, and I think that there's great potential here moving forward.
Okay, one last chance for questions. Any questions from the audience? Any questions online? Okay, fine. Michael, thank you very, very much. Excellent. What I'd like to do now is to call our next speaker up to the podium. He is Dr. Yasunori Aizawa, who is an associate professor of life science and engineering at Tokyo Institute of Technology, and he is CSO and co-founder of Logomix. So, Dr. Aizawa, if you would make a little bit longer introduction of yourself and then your presentation. Thank you.
Thank you very much, Dr. Pisano. My pronunciation is okay. Pisano-san. Yeah, and also thank you very much for the organizers for this great opportunity. Actually, this is my first time here, believe it or not. A very beautiful building, and I'm so envious. Yeah, and then this time the topic is bioproduction and also the big data. Actually, during my talk, I've never have any topics about big data and the bioproduction per se, but I hope you're gonna see that some kind of future of the bioproduction, because I'm talking about the human genome engineering, the engineering of the human iPS cells. And then in that field currently, it's probably more related to the pharmaceutical science like this place, right? But in near future, probably 10 years, 20 years from now, probably not only the human cells, but also any other higher organism cells or cell system or biological system could be useful for the bioproduction. So I'm seeing the future. I think we have to, and then one time, I don't remember when, but some past, I told myself that it was time for us to start breeding up some methods for the human genome engineering. So that I'm trying to show you our first chapter of the human genome engineering technologies we're developing in my laboratory and also in my company. So that the title is I changed the title a little bit: Academic and Industrial Potential of Large Scale and Precise Human Genome Engineering. So this is just introduction slide. So as you know, the genome technology revolution is ongoing right now, right? And then the human genome reading technology came first, right? And then right now, the DNA sequencing technology got mature, but still, I mean, the revolution is ongoing in the field. I think probably Hosokawa-san will touch some topic in his seminar today, I think. And also, as all of us are aware, right now, I think the genome editing is booming, right? Every issue, or every month, I should say, Nature, Science, or Cell, we see the new technologies based on the genome editing. But as you may know, aware, I think right now the genome writing technology is also coming. And then I'm mostly touching the genome writing technology, especially for the human genome. So today's agenda, so I start with just a quick introduction of genome writing, not editing, writing. And then I go through some genome engineering technologies we are developing in my lab and also in my company. And then I will touch the last some applications based on my some tools. All right, so start the genome writing history here. Sorry. Yep. So this is, I mean, as you know, the human genome, genome writing just started. Around 2000, the people started synthesizing the genomes of the virus or some phage because they're very small, right? And then, as you know, that around 2000, in 2008, Craig Venter actually published a paper about synthesizing the entire bacterial genome. But he did that first in vitro, and then he basically did it in vivo, in cells. And then in 2000, that's a 2010 paper, right? And then 2016, he minimized the bacterial genome, as you know, notes. So that during the time, or almost 8 years, only the Craig Venter is a person who is doing the genome writing. But in 2019, the Jason Chen at Cambridge, he synthesized the entire E. coli genome. His purpose was to record the codon. So he wanted to introduce some non-natural amino acid in a living cell system so that he basically first removed some codons and then from the genomes and also from the T cell, tRNAs of these codons also removed from the E. coli and make some vacant codon. And then he replaced that codon to encode non-natural amino acid in the E. coli. So this could be done only by genome synthesis, genome writing. This cannot be done by genome editing for sure. And then, as you may know, that around 2000, actually this year, 2023, we are going to finish up the synthesizing the entire yeast genome. So that this is this effort is done by like an international consortium called SC 2.0, Saccharomyces cerevisiae 2.0. And then the team was led by Professor Jeff Buka at NYU. And then the scientists from 6 countries get together to synthesize all the 16 chromosomes of the budding yeast. And then actually, I'm the person involved in the synthesizing the chromosome 4. Sorry. And then, yeah, if you're interested in the effort, please go to the bioRxiv paper you see below. And then I just want to just touch how we synthesize the genome. It's very simple. We just did stepwise extension of this DNA from shorter fragments into the long ones. So first, just synthesizing some 70 nucleotide single-strand DNA and make a complementary strands to the double-strand DNA, and then do basically PCR. We just assemble the fragment and then clone them into the E. coli. And then once we had the clones, we also extract some clones and then again, assemble them to make a longer fragment. And once we make the longer fragment, we use a fragment for swapping the endogenous genomic loci within the yeast cell from the telomere, one telomere to the other telomere, something like this. So very simple. I mean, it's simple, sounds simple, but it's not so easy. But we basically repeated this whole process to synthesize the entire yeast genome. And then this is the synthesizing part, right? And then this is the design part. We haven't synthesized the exact same wild type yeast genome. We changed many places on the genome. I'm not going to the detail. But we just deleted some introns, and then as you see bottom, we removed some virus-derived sequences, and also we substitute one stop codon to either of the other 2 stop codons to make one stop codon vacant, as the JSON did for the E. coli genome. And also we relocate the tRNA genes. As you know, the tRNAs are scattered several places on the yeast genome, but we just make them into one spot. And also the last one, we add log speed. I want to go to some detail about this. So we add log speeds everywhere, many places, I should say. So in this design, we introduced the log speed at the last part, the end of the non-essential genes in the yeast, which counts up like 5,500 genes. So that almost all of the yeast genes on this synthetic genome has a log speed at the last spot. And having these log speed sites, and then if you express the CRE, as you know, there's some recombination occurred, right? So that in some yeast cells, some genes, in this case, gene B will be deleted, or some gene will be duplicated, make two copies, something like this. So that this system is called a scramble system. And then the beauty of this method is that, of course, we are synthesizing one kind of yeast genome, but if you overexpress the CRE of the cell population, each of the cells in your tube have a different rearrangement of the genomes. So in the end, in one tube, you have many variety of yeast cells, and each of which has a different gene duplication or deletion. So that you can create some kind of, this is also platform, I should say. So we can create some libraries of yeast strain. And then this could be used for the screening, okay, to get some useful yeast strain, something like this. So this, again, cannot be done by genome editing, right? This could be done only by genome writing. So, and then I want to change a little bit. I want to shift the scope from the microorganisms to the human, the higher organisms. So as I mentioned at the beginning, I think in near future, we're going to face the era when the bioproduction or many synthetic biology fields get into the higher organisms. So we have to get ready for that. But the problem is that in comparison of the genomes of the E. coli or yeast, the human and the other higher organisms has a very tough genomes for engineering, right? So that the yeast and compare the human genome is gigantic, right? So we have to deal with a large target. And also another pain is that human genome has two copies of the genomes and genes, so biallelic cells. So this is also very tough part. And also in terms of design, we don't know much about the functionality of most of the non-coding regions in the human genomes. So that we have to have some method also for the better design, not only for the reconstruction, but also the designing of the genomes. So that from here, I'm shifted to the second topic. So with knowing, okay, the human genome is tough target for the genome engineering, but we have to get in the field. So that you have to get ready for that. So that to do so, we, for the last many years, 5 or 6, 7 years in my laboratory, and also 3 years in my company, the Logomix. So we are trying to get ready for the timing. So these are tools we developed for human genome engineering. Two for designing, two for rebuilding of the genomes. So I'm just touching the surfaces of these tools. First, the DEGES. So DEGES stands for the Digital Evaluation of Genome Essentiality for Cell Survival. So literally, okay, this method tells us that which genomic region could be important, essential for the cell survival. In other words, which genomic region shouldn't be removed if we want to keep the cell alive. So, and then the method is as follows. Very simple, actually. So we use a paired guide RNA to chop off the target region, in this case in green. So sorry, the slide is really small. And then just we let the cells grow after we introduce the 2 guide RNAs. And then just we let the cells grow, and then we extract the genomic DNA at a certain point, okay? And then these genomic DNA are used as a template for the PCR, junction PCR. And for the junction PCR, we use a primer targeting in this case, the green region, okay, to measure the ratio, in this case, the ratio of the blue cells out of the all cells, okay. In the blue cells, the target region is successfully removed by the paired guide RNA. In the red cells, the target region are still there. The deletion cannot be not happened here. Okay, and then our target in this case, the green region is very big, so that the efficiency of the removal of this long region by paired guide RNA is very low. So the most of the cells are red, but we want to measure. But as you know, that our target is very long, so that only the DNA extracted from blue cells can be amplified. Okay, so that based on this principle, okay, we measure the ratio of the blue cells, and then if you see the decline of the ratio of the blue cells along the cell culture, meaning the deleted region is important for cell survival. But if you see the blue cell ratio constant along the time course, meaning the region can be removed without affecting on the cell survival, cell growth, right? Very simple. And then we apply, so this is very important. Okay, I want to just recall that this method is for the designing. So which part could be removed, which part of the genomic human genome can be touched. Okay. All right, and then we used this method to ask this question: how much can we trim genomes? And we targeted 1% of the human genome, which is like 33.5 megabases, which is close to the telomic region on the long arm of X chromosome. Okay, 1% human genome minimization project in my laboratory. And then this region includes like 200 protein genes. It's the same, very normal. And then to do this, we want to evaluate the methodology of the DGES. And also, we wanted to have some rough idea about human genome design rule. I want to show you just the data. So this 1% human genome region includes 8 non-essential genes. So that if even one of these genes will be removed, cells are going to die, okay? It's non-essential genes. So, and then we target each of the 8 essential gene loci by using DGES. And as we expected, the ratio of the blue cells are going to down, right? So the very beautiful data. So this shows that our method is working. And then we target the region between the non-essential gene region, in this case 4 in blue. So yeah, and then this is the data. For example, R1, this region includes like 36 genes, but each of them are non-essential genes, okay? And then we can remove the 36 gene at the same time without losing the cell viability. So we can see the same thing for R2, but we see the decline of the blue cells if we remove the R3 and R4, meaning this region could have some genomic region which is essential for the cell survival. And then we just narrowing down, okay? We repeated the DGES experiment to the R3 and R4 region. And then we found that some new essential genes. This is kind of unexpected result. I expect more interesting, the surprising data, but it seems like people just missed essential genes in this cell we used. We found the same result from R4 region. So yeah, but from here, we are sure that the DGES is very useful tool for the human genome designing. And also about the human genome design rule thing, the human genome can be trimmed without losing cell viability. We didn't expect we can remove such a long, many genes at the same time, but we observed, we made such observation. So this is very important for the human genome writing. Okay, so and then I just want to touch the upload, which is just one slide. So yeah, as I mentioned, that so upload is like a platform to synthesize a long DNA of any sequence you want. Okay, and then this platform is from my experience is from through the SC 2.0 activity. I don't want to go to the details of the platform here. And I move to the last one, the UCIS. This is a center tool for our human genome engineering platform. So UCIS stands for the Universal Knock-in System. So this is basically, as so speak, I mean, this is a method for knock-in of the any long DNA into some specific region in a human genomes. So we can target like up to the 10 megabase region. And also we can do the allele-specific modification, as you see in this picture, from two red alleles into the one yellow and one green sequences in each allele. And also this method is scarless, meaning you don't have to leave any unnecessary sequence at the target region. So very clean. So this is very important to make multiple round of genome modification at a different location in the same cell line. So this is very essential. And also efficient, very speed, efficiency is very high with a hand. So if you're interested in this method, please go to this paper and also this patent too. And then, but the one drawback of this method is that after the change, we have to have the sequence which is less than 50 kb. This limitation is comes from just the low efficiency of the low efficiency in the integration of the long DNA into the iPS nuclei. So, but as I showed you in the DEGES, so many genomic regions can be trimmed without losing functionality. And then the data from DEGES is accumulated in a different platform called Gemini, which is algorithm for the minimization of human genome. So that this whole process is connected to each other. Each tools are connected to each other to accomplish the genome engineering in the human cells. So probably I have to rush. So this is just a scheme of the UCIS. It's a two-step homologous recombination based, and we use CRISPR, and also we use a 2 drug selection marker for biallelic modification. So we combine several pre-existing genomic engineering tools into one platform to make this whole process. Yeah, so I just want to go through. The last one, probably this would be the most attractive, probably today's most audiences. The application. I give you some 3 applications of our method. First, this is more like academic application of UCIS. So as I showed you that the UCIS allows us for the very precise scarless biallelic human genome modification in cells. So that we can do like a single nucleotide resolution mutagenesis efficiently. So and then so that with this methodology, we wanted to tackle a very basic question, biological question, what intron does. So as we all learn in undergrad or even in high school, so there's some called intron sequence, right? And then we learn that. I teach that in the class, but actually we don't know much about what intron does. So we wanted to do a very direct mutagenesis to just remove the introns from the genes on the chromosome in the living cells, and then to see the effect. We can do that. So we, in this paper, we target the 3 different genes. Sorry, it's very small. So this is gene CD44, which is a 94 kilobases long, and which has like roughly 20 introns. And we remove these introns from the endogenous chromosomal loci to make an intron-free CD44. And we did the same thing for the MET, which is a 126 kilobases long, and also the APP, this is 290 kilobases long. So we remove the all introns from genes, both alleles. And then, as you saw, and then we check the expression. So to test the intron is required for the expression of the genes. And then for the CD44, we see some reduction of the expression, but still the transcription is almost the same. But in the case of MET, the intron is necessary. If you remove all introns, the transcriptions are gone completely. And then for APP, we still see the expression, but see the drastic reduction in expression. So that the answer is like a gene, the intron requirement is different from genes to genes. Maybe we knew this conclusion before this experiment, but seeing better, right? So yeah, this is the thing. So we still, but this is very important for genome designing again. We don't know much about the intron design rules in human genomes, human cells. The second application are more related to the some therapeutics. So we use this method to create isogenic iPS cells modeling disease. So as you probably, as most of you guys noticed that these days people use iPS cells from patient and also iPS cells from healthy donor to compare them to see the mechanism of the disease and also use the pairs for drug screening, right? But the problem is that these cells are from different people. So the genetic background is different. So that could be a noise for the screening process. So instead, we introduce mutations in healthy iPS cells, and then the genetic, in this case, genetic background is same, so that we can see the real effect of the mutations we introduced. And then in our case, we don't want to deal with some just a disease which could be caused by just a single nucleotide mutation, right? So that we want to have more, we want to target the disease which could be caused by a little more complex mutations. So our first target was like a CAG repeat disease, so Huntington. So as you know, the patient, severe patient has an extension of triplet repeats, CAG repeats at the gene called Huntington. So we introduce some particular repeat number on one allele or both alleles in a healthy iPS donor to create these 5 isogenic iPS line modeling Huntington diseases. The first one is the normal cell, both allele has 10 repeats, and then the second and third one has only one allele has extension. Second one is 55 repeat, and the third one is 99 repeats. And then, as you know, this disease is a dominant disease. So the patient has the extension only one allele. But with our method, we can create some artificial iPS cells modeling Huntington disease, which has extension on both alleles. We expected these cells has a severe symptom, and that could be useful for the drug selection, drug screening, and also the studying the disease mechanisms. Yeah, so you cannot get these bottom 2 cell lines from the patient practically. And in my startup, we expanding the lineups of this isogenic iPS cell modeling CAG triplet repeats, as you see here. So if you're interested in using these kind of cell lines, please let us know. And the third one, sorry for taking a long time. So the therapeutic iPS cells. So this is a kind of another small platform to make some multi-functionalized cell modality that could be used for the cell therapy or regenerative medicine. I'm not going to the detail because the time is over almost. But I just want to introduce, briefly. So we called hypoimmunity control technology. So we first create some universal donor cell of iPS cells. But in this case, our method is a little bit different from others. Instead of knocking out B2M or C2TA, which are important for the expression, HA class 1 and class 2 gene expression respectively, we just remove all HA class 1 genes on the top one, and also we remove not only class 1 genes, but also class 2 genes from iPS cells. In total, for the second one, we removed 2 megabases. cytospecifically from both areas. So, and then we can also introduce our UCIS landing pad, where you can insert a large gene cluster efficiently. So this is UCIS now. And then lastly, I just want to show some data to show this method. So we introduce, so first we removed all HLA genes, right? And then we are trying to get some specific HLA haplotype gene back to the cell. And then so we introduced HLA A24201 and also B721. So in this case, we not only introduced the protein coding region of HLA, but also we also transfer endogenous promoter, downstream sequence, and introns. So that way, we can have the very natural expression of HLA genes in a cell. And then this is the data. So we first used some iPS cells, which has expression of B7, A24, A2. And after removal of HLA genes, these expression gone, the second line. And the third one, so we reintroduced A24 and also B7. And then we see the getting back, the expression of getting back. Yeah, sorry for rushing the last part, but this is a summary. So I just gave you the quick introduction of gene writing and then showing some genomic tools we created. And then we share some examples of our applications. Thank you very much for your attention.
Okay, Aoizawa Sensei, thank you very much again. And I want to again turn to the audience, either in person or remotely. Are there any questions? We have a few minutes for questions. Are there any questions online? Nothing. Any questions here in the audience? So while you have one. All right.
Hi, thank you. That was very nice. I'm very excited about this technology, especially the genome writing. I can see lots of applications in heterologous production and the use in biotransformations. But when it comes to your yeast project in particular, how are you dealing with the mitochondrial DNA?
Intact. No change.
No change.
No change. But I think some researchers are working on that, right? Yeah, I know that could have a huge impact on the cell metabolism, of course. Yeah.
Okay, follow-on questions? Anybody else from the audience? Anybody online?
Yeah, one from online. I'm not familiar with this genome writing tech. Can this tech be applied to produce proteins that are deficient to cure diseases, such as hereditary disease?
Hereditary disease. Yeah. Well, generating. Yeah. Thank you very much for the question. Probably just synthesizing to produce some specific proteins. Maybe the generating is not necessarily useful. Maybe many other tools will be useful. But the point of the generating is that you can change the metabolic pathway to network in the host cells for the bioproduction by using genome writing. The genome editing, with genome editing, maybe you cannot change the entire, it's like a landscape of the some cell systems in the host cells, right? So if your, for example, proteins are very deleterious, it's a poison to your host cells, probably changing some host cell genomic region could be useful to increase the expression of the protein you're interested. But yeah, could be. Sorry. I hope I am answering the question. Yeah.
Okay, so in the interest of time, I think we're at time.
Thank you.
Aizawa Sensei, thank you very, very much.
Thank you very much.
Okay, in the last 15 minutes of session one, we have a video. Professor Bernard Paulson is unable to travel. And so he has made a short recording of his presentation. So he offers his apology that he cannot be active online. And if we can, could we play the video now?
Hello, this is Bernard Paulson. I'm a professor of bioengineering at the University of California in San Diego. We all hear much talk about sustainability these days, and we know that there are biological solutions that help out with developing sustainable lifestyles. Therefore, there is much talk about biosustainability and biomanufacturing. I have been involved for now over 10 years in building an international research center to meet the goals of biosustainability. I would like to share with you today some of the lessons we have learned from this undertaking. I have run the Systems Biology Research Group at the Jacobs School of Engineering for 25 years now. I'll be happy to tell you a little bit about our status and our capabilities. Before I start, I will point out that we have a YouTube channel where we post our courses, educational materials, as well as research talks. So today, I would like to divide my talk into two parts. One is the need for biofoundries, which is a near-term goal, and then a new discipline of genome engineering, which is a longer-term goal. Biofoundries are in some way similar to the old-style mechanical shops or electronic shops at universities, in engineering schools, which are infrastructures that allow you to build things. A biofoundry is shown in this slide as a part of a larger process. It's an iterative process of designing, building, testing, and learning. And I will describe biofoundries in detail later on. The users are design teams that are designing biological constructs for different applications, as shown. That could be sustainable chemical production, natural products, or microbial foods. And since all of these are very applied undertakings, a translational outlet needs to be provided to help products get into the market. The center that I mentioned earlier is called the Center for Biosustainability, funded by the Novo Nordisk Foundation in Denmark. It was an international center that has locations in different countries across the world: Denmark, Sweden, here in California, operations at Kaestin, South Korea, and other places. It was established in 2011. It grew very rapidly to 300 FTEs and now occupies this beautiful 12,000 square meter building, published 1,700 papers, raised over $500 million in research funding, spun out many companies. Overall, been a very successful center and was well ahead of its time. Biofoundries are now quite popular, and the White House in the United States has put out a call for building biomanufacturing and industrial biotechnology in the United States. That is outlined in the document shown, not yet been funded by the US Congress, but industry would like to have biofoundries in the four corners of the country, mostly focused on scale-up and manufacturing technology. The products that come out of industrial biotechnology and medical biotechnology are shown here. We are, of course, very familiar with vaccines and antibiotics and therapeutics. We are now focusing on microbiomes, the environment, and various food products shown in this slide. A biofoundry is comprised of 4 parts. First, there is the design step. After the design step, you build the construct, then you take it into testing, and then we often get test results that are different than what we designed for, so we try to learn from the test results and try to figure out why the design didn't produce what we wanted to. So this is an iterative process. Above the diagonal line shown from top left to bottom right, you see a separation into the 2 parts of the loop that are experimental and those that are computational. Below this diagonal line, we have experimental work, mostly comprised of technologies that are widely available, but there are some specialized capabilities in these 2 steps of the loop that are maybe particular to a university or a department. Above this line, we have the learn and the design step. These steps are primarily done in a computer using databases, data analytics, algorithms, and computational biology. Currently, there is less sophistication in these 2 steps than the experimental ones. So one of the limiting factors in developing biofoundries is now to perfect these 2 steps. And this is the domain of engineering, computation, and data science. There are many different technologies found in each one of these quadrants. And here we illustrate some of the strengths we have at UCSD. One of them is that we have an automated laboratory evolution machine that helps us build designs biologically, so not by direct intervention of DNA. And above that diagonal line I talked about before, we have a variety of computational tools and data analytic tools that are mentioned in here. And as I mentioned also earlier in the talk, these are readily available in the public domain. And in fact, some of them are used on a worldwide basis. In particular, our technology with analyzing metabolic networks to assess metabolic functions and productivity of organisms is in worldwide use and has been so for probably almost 20 years now. Here is an example of automation that we are building. So here is a robot, a liquid handling robot that carries out the laboratory evolution. So you start maybe out with a strain that does not tolerate the product very well, and you have it put it through an adaptive evolution process and allow it to mutate and generate a tolerance. Sophisticated software is built for devices like this to control the instrument, collect the data, archive the data, and then subsequently analyze the data. This is a big part of biofoundries in the future, laboratory automation. Here's an example of how a device like this works. You serially passage an organism on a particular substrate. And you can see in the middle panels how growth improves over time as mutations are acquired. And then in the panels to the right, you see a genetic map, which shows where you find these mutations. And in the bottom right-hand corner, you see some biochemistry that shows the proteins that have mutated to give the organism better performance. So this is a short introduction to biofoundries, the technologies they contain, their role, and some of the special advantages we have here at UCSD. For the second half of my talk, I'd like to speak to a longer-term goal which is the establishment of a new discipline called genome engineering. And yes, we will probably soon be designing small genomes de novo or by biomimicry to generate cells that have capabilities not found in nature. I should say right away that such organisms are highly fragile and will only survive under the biomanufacturing conditions that you have designed. Here is a broad perspective of the history of biology. To the far right, you see dates in the mid-1800s when Darwin developed his theory of evolution. And roughly in the middle of the diagram, you see around 2000, there was a convergence of technology developments that allowed us to study genomes as a whole. So in the late 90s, the first full genome sequences of industrial microorganisms became available. We started building genome scale models around 2000 here in San Diego. Whole genome synthesis happened then about 10 years later. And now we have a large number of machine learning methods to analyze and model biological data. Somewhere roughly, say, 10 years ago or so, biology bifurcated into being both a scientific discipline, as is stated under my image on the slide, and an engineering discipline. So synthetic biology came along, and we were able to edit and manipulate genomes to create new functions. And this forms a basis, in my view, for a department or new departments of genome engineering that most likely will appear before the end of the century, before the end of the decade. Such a new discipline will be built on certain fundamentals, just like other engineering disciplines are. First, we need to know something about the object we are designing, genomes, and whole genome sequencing has helped us to understand genomes better by simply sequencing thousands, if not tens of thousands of them. And this was enabled by a company here in San Diego called Illumina that dropped the cost of sequencing massively by about 3 logs in the late 2000s. So throughout the 2010s, there was an exponential growth in the availability of new genome sequences. Unfortunately, that curve is right behind my image on this on the screen right now. The second technological development was the development of genome-wide measurements. So we could look at genomes not just as objects, but we could also study their state on a genome-wide basis. And as shown in the insert in the middle of the bottom of the slide, we can now map dozens of different data types against the genome sequence to understand the state, the functional state of a genome in a given environment. Number 3, we need to be able to build. And in the top right corner, you see the Nobel laureates for CRISPRs, which was a revolution in genome editing. In the bottom left corner, you see achievements in building large-scale DNA constructs up to whole bacterial genomes and even whole chromosomes in eukaryotes. So we are now able to build very large DNA constructs. And pictured in the very corner to the far left corner of this slide is a face shot of Craig Venter that drove this forward, and his institute and company that has worked in this area is located here in San Diego as well. The fourth component of an engineering discipline is analysis, both statistical and mechanistic models. And those are shown in the center top of the slide. And this is a field that's also been pioneered here in San Diego over the last 20 years. So you see San Diego and the larger San Diego community has worked diligently over the last 10 years to put the fundamentals of this discipline in place. Just briefly on the computational tools, as in all engineering, first principles-based models or mechanistic models are used. But the truth be told, a lot of engineering is based on empirical correlations, which are based on statistics. So shown to the left in this diagram is a panel showing a series of different data analytics that have been developed lately. And they are knowledge-based and interoperable, and those are likely to have a revolutionary impact on the development of biology during this decade. To the right of this slide, we show more classical computational tools that are based on first principles, things like thermodynamics, sequence analysis, kinetic modeling, and metabolic flux modeling. So this capability is unique to engineering and data science. And this is the component of the biofoundries that I mentioned earlier that is least well developed. With the advancement in dealing with DNA, building it, sequencing it, analyzing it, and so forth, we now see the possibility of a whole design loop that is all based on DNA technologies. So you can design and build your DNA constructs based on genomic information. But then, of course, you have to do the physical embodiment of those genomes and build an organism and test it. But we believe that by the late 20s, this will have reached the level of sophistication where we can contemplate basically designing genomes from first principles. So here are the key takeaways of my talk. So genomes are a fundamental construct of nature, and we will build an engineering discipline around them, just as we have for electrons, chemicals, mechanics in the past. These departments are likely to emerge during this decade at leading engineering schools, and is driven by a culmination or convergence of the key technological drivers that I mentioned just a minute ago. To enable this vision, such departments have to have biofoundries, and I described them in detail earlier. And there is, of course, precedent for building analogous operations in engineering departments in the past. There are some differences here. They are big and expensive and costly to operate. So support for them has to probably come from federal agencies and or industry. They are likely to be regional resources, and some will become national resources in the United States and potentially international resources. And they will serve both academic research, basic research, as well as translational research and company formation. Most of them will have to have a pre-pilot capability where you can actually prototype industrial processes, maybe up to 100-liter scale or so, and include product recovery. And that is already a proven concept through the center that I introduced you to earlier. Many of the companies that came out of that center went through that pre-pilot plant before getting into the commercial arena. And finally, I will re-emphasize that data generation, high-quality data generation, and data analytics are now a key driver going forward, as this is one of the components that limits progress in the field. And with that, I will stop my talk. And hopefully you enjoyed this short overview of biofoundries and genome engineering. And I gave you some glimpses of the products that this technology is likely to generate and support production of in the future. Thank you very much for your attention.
はい、皆さん、いろいろとトラブルがあってですね、誠に恐縮ですが、第2部の方に移らさせていただきます。今ですね、第1部、プラットフォームテクノロジー、と題してですね、いろいろと素晴らしい最先端の取り組みのプレゼンテーションをしていただきました。で、続いてですね、第2部、こちらはですね、グローバルインダストリートレンズということで、お三方から20分ずつ、プレゼンテーションをしていただきます。まず最初の、プレゼンターですが、ボストンコンサルティンググループ、マネージングディレクターであり、アンドパートナーの柳本さんからですね、グローバルインダストリアルトレンズ in バイオマニュファクチャリング R&D ということで、柳本さんはですね、ちょっと体調不良の中をしてですね、リモートでプレゼンテーションをしていただけるということになっております。柳本さん、聞こえますか？映るかな？
はい、聞こえております。
はい、じゃあ、ご無理のない範囲でぜひよろしくお願いいたします。
どうぞよろしくお願いいたします。画面見えておりますでしょうか？
聞こえてます。画面も見えてます。
はい、よかったです。ちょっとうかつにもコロナにかかってしまい、鼻声で大変失礼いたします。で、現地でも参加できなかったこと大変申し訳ございません。で、今、須山さんからご案内があった通りですね、ここまで、かなり、技術的なところで、深く、議論をされてきたことと思います。で、この後は第2部というところで、インダストリー側の取り組みを紹介する、その最初のセッションということで、個別の、企業における取り組みではなく、インダストリー全体として、このバイオマニュファクチャリングというのはどういうものかと、どう捉えるべきかということを話せればなというように思っております。具体的にはですね、まず、インダストリーにおける R&D のトレンドをモダリティの観点から少し考察した上でですね、その中でも特に新規モダリティの中のセル&ジーンセラピー、細胞遺伝子治療といったところでのマニュファクチャリング上のチャレンジについて言及し、最後にですね、それらの、マニュファクチャリング上のチャレンジも含めてですね、ビジネスとしての追い風と向かい風というものを少しお話しして、次の各インダストリーの代表の方にバトンをお渡しできればなというように思ってございます。よろしくお願いいたします。はい。で、まずは R&D ですね。はい。ここ皆さんご存知の通りというところです。一番、左にスモールモリキュール、低分子があり、組み換えがあり、抗体があって、でそこから、ADC、CAR-T、ジーンセラピーといったような、いわゆる、新規モダリティというものが始まり、で、今最もホットな mRNA ですとか、あとは、どんどん出てきているオンコリックバイラスとかいうものがあり、これから、世に問うていかれるであろうモダリティが続々とこの後並んでいるというところ。で、バイオマニュファクチャリングと一言でくくったときに、もちろんここからここまで全てバイオマニュファクチャリングではありますが、おそらく皆さんが今日、この前もですし、この後も議論されるのは、ここからここのいわゆる新規モダリティというところかと思っています。で、特に私はこの後、このセル&ジーンセラピーというところに少し焦点を当てたいなと思っております。で、今の、R&D のトレンドというところが、パイプラインにどう跳ねているかと。で、結果としてどういった市場になるかというのを、がんの、オンコロジードラッグの薬、市場において見てみたいと思っております。で、一番下から、低分子、コンベンショナルバイオロジクス、いわゆる抗体ですね。でそれから、いわゆる新規モダリティということで ADC、バイスペーシフィックからセル&ジーンセラピーというところになります。で、ご覧いただいてお分かりになります通りですね、低分子、2021年、今現在でいうと55%マジョリティを占めており、で続いて、抗体が38%、この2つで、9割を超えている状況。これが2028年には、先ほどご覧いただいたモダリティの多様化がさらに、進展してというところで、20% 後半はこの新規モダリティが占めるということになっております。でそれぞれ経側で示したときには、やはり、セルセラピーというものが一番高い経側を示しているということになります。ただ、一方で、特筆するべき点としては、そのような状況でありながらも、依然、低分子は伸びており、 まだ 40% は5年後の時点でも市場を占めているというところですね。ですので、新規モダリティ、当然これから、伸びてきますが、新規モダリティが全てではなく、低分子であったり、いわゆるコンベンショナルな抗体というもの、これらが依然市場の中心であることに変わりはないというところ、これは特筆すべきかと思っております。で、その新規モダリティの中で、各社がどのように注力をしているかということになります。で、上段が、グローバルのトップ10、下段が日本のトップ10ということになります。で、各社はここに現れている以外にもですね、取り組まれていることというのは、多くあるというふうに、存じておりますが、あくまで、公表データベースというところで、集計してございます。で、こういうところがより数が多いところというところになりまして、ご覧の通りですね、各社、新規モダリティといっても、注力している場所は様々でございます。例えば、ロシですと、バイスペシフィック。BMS ですと、ジーンモディファイドセルセラピー。で、片やアストラゼネカですと、DNA、RNA というところ。このあたりが、かなり、各社において注力している対象が違うということが見て取れるかなと思っております。一方で、日経に関してで言うと、比較的、セルセラピーというところは各社手掛けていることが多いというところがあり、それに加えてというところで、バイスペシフィックですとか、DNA、RNA というところに広げているというようなことが、比較的見て取れるのかなというように思っております。というところまでが、R&D のトレンド、簡単に見させていただきました。で、この後、マニファクチャリングの方に入るんですけれども、特に冒頭申した通り、セル&ジーンセラピーに注目するとしたときに、とはいえ、セル&ジーンセラピーにおいても多様な治療法があるというところで、少しカテゴリーを分けてみたいと思っております。で、まずジーンセラピーに関しては、インビボとエクスビボに分かれると。で、セルセラピーに関しては、自家と他家に分かれるとしたときに、サプライチェーンですとかマニファクチャリング上で言うと、真ん中の2つというのは、ほぼ同じような、もちろん中で使われる技術は、大きく異なるんですけれども、プロセス上は、それほど変わらないというところで、この2つがくくれるような形になっております。で、特にちょっとここを、深掘りして見ていければなというように思ってございます。はい。で、こちらはですね、マニファクチャリングを中心とした、先ほど申し上げた、自家の細胞治療、もしくは、イクスビボの遺伝子治療のプロセスになります。細胞の採取に始まって輸送して、細胞を加工して、分析、検品した上で輸送して患者に、医療機関に戻し、患者に戻されるというところ。この一連のプロセス。これ自体はこの時点ですでにこれまでの製薬企業のサプライチェーンとは大きく異なると。もともと製薬企業の場合は当たり前ですけども、工場から始まって、川上から川下にきれいに流れていくようなサプライチェーンなところが、ご覧の通り、患者に始まって患者に終わる、医療機関に始まって医療機関に終わるというような、行って帰ってのプロセス、これを管理しないといけないというところが、まず一つ難しいところになりますと。で、その中で、マニファクチャリングというものに、目を向けたときにも、この採取のところから、遺伝子導入した上で、分離を進めて、で、また、凍結して運べる状態にするというところ、このプロセスにおいても、今の、医薬品とは全く異なるプロセスが求められるというところで、もう同じ、医薬品のカテゴリーに入るような製品であったとしても、このサプライチェーン、マニファクチャリング上で求められるプロセス、技術というものが全く別物だというところ。これがなかなかですね、もちろん今日お集まりの皆さんというのは、よく、ご理解されている皆さんだというように思いますけれども、経営の皆さんですとか、サプライチェーン、マニファクチャリング外の皆さんには、理解がなかなか、及んでおらず、数年前に CAR-T が同時に上市した際でもですね、 やはりこの一番のネック、もちろん、表立っては薬価どうするかというところが大きな話題になってましたけれども、オペレーション上では、このサプライチェーンをどうするのか、どう CDMO を育てていくのか、確保するのかといったところからが一番の、ボトルネックになっており、それに必要な IT システムをどう導入するのかというところが一番各社不信されていたところかなというように捉えております。で、特に今申し上げたエクスビボのジーンセラピーと、自家のセルセラピーの難しいところというところをマニファクチャリング上の観点で4点挙げております。1つ目がですね、やはり自動化が難しいというところ、特に最初の世代に関しては、基本マニュアルで始まってくるというところで、自動化できないというところが一番難しいところ。で、だからこそというところで、スケールアップがとてもコストがかかると。で、その自動化できない部分っていうものが多々残る中で、職人技、ある人に言わせると、日本酒作りの杜氏と一緒だというところですね。タンクの中で何が起こっているかをタンクを見なくても分かるというような、そういうような技術まで求められるということもおっしゃる方もいらっしゃいますけれども、それぐらいの、職人技も求められて、スケールアップが難しいというところ。でさらにはですね、やはり変動要素、不確定要素が大きいというところで、科学的に合成したものではなく、細胞という一つ一つの特性が異なるものを相手にしているがために、品質が安定しないというところが、3つ目のチャレンジですね。で最後は先ほど申しました通りですね、自社だけではやりきれない中で、他社に頼らざるを得ないにもかかわらず、特に日本においてはですね、この CMO、CDMO というもの、この新規モダリティにおける CDMO が発達していないというところが、大きなチャレンジになってくるというように捉えております。でこちらは参考までにですけれども、主要な、CGT プレイヤーにおいてですね、すべて、インハウスで作っているのか、すべてアウトソースしているのか、選択的にアウトソースしているのかというものを分けたものになります。ご覧の通り、すべてアウトソースというものはないものの、一方ですべてインソースというのは、マジョリティではなく、一番多いパターンというのは、外注と組み合わせているというパターンになりますので、先ほど申した通りの、外部活用の難しさ、特に日本における CDMO、CMO の不足というものは顕著に出てくるところになってくるかと思っております。ちょっとここは飛ばさせていただきまして、で今までのところが、マニファクチャリングにおける、チャレンジというものを簡単に紹介させていただきました。でおそらくその点に関しましてはですね、この後の、お二人の演者の方が詳しくそれぞれの企業における取り組みを話してくださるものと思いますので、最後私の方からはですね、さらに一歩引いてですね、事業全体として捉えたときにということのお話をさせていただきたいと思っております。で、こちらは、これからマーケットがどうなっていくかというところで、左側がセルセラピー、右側がジーンセラピーで、ご覧の通り、セルセラピーの方は 56% の経過で3兆円ほどの規模。で、ジーンセラピーに関しては7割を超える経過で、2兆円弱の規模まで成長するということが、予想されています。で、ただですね、そこに至るには、やはり、まだまだ向かい風が多いというところになります。一部先ほどのマニファクチャリング上のチャレンジともかぶりますけれども、1点目はですね、スクルーティニーというところで、やはり、もともとその期待されている治療であるということ、加えてですね、高い薬価というところで、社会からかなり注目されていて、かなり、その注目を浴びた状況下で、開発を進め、社会的にそれが、妥当なのかということを精査されるというような、そういうようなチャレンジがあるというところ。でスケーラビリティに関しては、先ほど申し上げたように、主にマニファクチャリング上のチャレンジですね。で、あとは、3つ目のところはコンペティティブネスというところで、全ページの通りの市場の伸びですし、あと冒頭申し上げたような 皆が、こぞって研究している領域ということにもなりますので、これまで以上に、競争が激しくなってくると。この中で、これまでの、コンベンショナルなモダリティと同じように、ファーストトゥーマーケットでいくのか、ベストインクラスでいくのかというような選択もこれからは迫られることになってくると。今までのように全てが全て、ブレイクスルーセラピーでファーストインクラスでということができないような時代もやってくるということが挙げれるかと思っています。で、最後はですね、プロフィッタビリティというところでですね、そのマニファクチャリング上のチャレンジで、もともとの、その原料単価の高さというところもあってですね、製造原価の高さということもあってですね、なかなか収益性が生みにくいと。でしかも薬価も高くなってしまうので、患者、ターゲットポピュレーションもなかなか広がらないという中で、いかに収益を上げていけるのか、いかにビジネスとして、大きくしていけるのかというのが、実は、このセル&ジーンセラピーの事業化における一番のチャレンジではないかというように考えております。ここはまだ、社会としても個別企業としても明確に解がないところで、ごく限られた先行している治療に関しては一定以上のプロフィタビリティを得られてますけれども、これからどんどん増えてくる治療薬、治療法に関して同じように収益性が担保しきれるか、もしくは収益性を担保した上で一定以上のビジネスにできるかというのは、まだまだ予断を許さないところかなというように思っております。で、とはいえですね、もちろん追い風は多々ありますというところになります。やはり一番はですね、患者のアウトカムが圧倒的に高いものが期待できるというものですね。で、だからこそ、ここにいる皆さんもそうですし、社会が注目して、時間とお金をかけて取り組んでいるのかというように思っていますし、でそれがゆえにですね、ペイシェントのアクセプタンスも非常に高いというところが挙げられるかと思っております。で、さらにはですね、そういう、アウトカム、社会的共生の高さというところからですね、レギュラトリーボディも一生懸命この領域に対して、遅れを取ることなく規制を変革していこうと取り組んでいるのが、世界的な情勢かと思いますし、それは再生医療三法に代表されるような日本におけるPMDA の取り組みにもつながっているのかなというように思っています。で、あとはですね、まさにここにいる皆さんの多くが日々取り組まれているように、これからさらに、科学技術が進歩してくるという中で、これ前ページ、もしくは前段で挙げていたようなチャレンジという部分が、これから、どんどん、解決していくだろうと。さらには、その患者アウトカムというところもさらに向上していくだろうというところ、この4つが、一方でというところの、追い風として、この市場の成長、もしくはこの治療の普及に望みを託せるような状況になっているのかなというように思っております。簡単ではございましたが、最後少し締めということでですね、R&D のトレンドからマニファクチャリング上のチャレンジ、簡単にではございましたけれども、概観させていただきました。でそれも踏まえて、この、新しいモダリティにおける事業化の難しさと追い風というものも、一緒に見させていただきました。で、やはりですね、何よりですね、この治療、特にセル&ジンセラピーというところを挙げさせていただきましたけれども、患者のため、社会のためにとてもなくてはならない、これから不可欠になっていくものだというように理解をしておりますので、ここでの本日の議論というものが、その一助になればなというように思っておりますし、この後の、残る2セッション、プラスパネルディスカッションというものが、皆さんにとって実りのあるものになると良いなというように思っております。はい、私からは以上でございます。
はい、柳本さん、ありがとうございました。グローバルな視点からですね、社会的な課題も含め、かつそれをビジネスの、側面から、も見ていただいてですね、本当に貴重なプレゼンテーションありがとうございました。お時間ちょっとありますが、どなたか会場の方、オンラインの方、ご質問等終わりでしょうか。いかがですか。大丈夫ですか。どなたかいらっしゃいますか。オンラインの方はどうですか。 大丈夫。大丈夫だそうです。大丈夫ですか？よろしいですか？じゃあ、井出さんどうぞ。
すいません。素晴らしいご講演ありがとうございました。2点お伺いしたいと思っていまして、先ほどその CMO とかいろいろその海外で、いろいろセル&Gに関しては、かなり多様なプレイヤーがいらっしゃると。で、その中で製造っていうものに対して、特に病院施設で最近製造施設を置かれるようなところが多いと思ってるんですけれども、そこの、参入に関して、柳本さんはどのように今後伸びていかれるかどうかというお考えを伺いたいのが1点目です。で、あともう一つ、低分子に関して、CG も8% ですか、伸びるとなってたんですけど、あれは、ジェネリックを含めての話なのか、それともやはり、その新規な、高度な低分子を含めてのお話なのか、その辺の、なんて言うんでしょう、割合といいますか、見立てをちょっとお伺いできればなと思います。よろしくお願いいたします。
はい、ご質問ありがとうございます。ちなみに、1点目の質問、少し確認させていただきたいんですけど、病院における、製造設備を、なんですかね、設置することへの参入でしょうか。何に対する参入
そうですね。
はい、
そうです。病院施設、ちょっとここはあまり私も詳しくはないんですけれど、
はい、
病院施設側がそのものが持ったり、あるいはそのベンチャーや企業が参入したりっていうことが、多様にあるんだと思うんですけれども、その、大きなトレンドとして、このどのような仕組みになっているのかっていうのをちょっと外観知りたいなというのが質問の趣旨です。
はい。どうもありがとうございます。最新の数値までは把握はしてないんですけれども、おっしゃる通り、病院側で一定の、設備を持つということは、すでに、起こっておりますし、これからも続くと思っております。ただ、細胞加工には一定以上のその設備、それは、規模も含めてですね、必要になってくることも多いというところですし、作る細胞によって、ラインもつどつど、組み換えだったり、その洗浄もしていかないといけないことを考えたときに、その治療法が単一の場合は、病院の中で、賄いきれるんだと思うんですけども、それが複数になったときには、おそらく病院の中で賄いきるだけの規模の病院というものが、それほど多くないと思われることから、院内には、一定、院内のものは普及しつつですけれども、規模が大きくなればなるほど、マジョリティは院外での施設ということになるのかなと思っております。ただ、やはり、施設に近い方が、その輸送の面でも有利になりますので、日本なんかですと、それほど何拠点もっていうことにはならないでしょうけれども、アメリカなんかですと、おそらく、地域ごとにですし、中国なんかはもう同じく地域ごとに、国がセンターを作ろうとしたりとかしますので、そういう形で、病院内のものと院外のものとっていうところで、住み分けが行われてくるんじゃないかなというように考えております。
ありがとうございます。あ
で
と、もう1点、低分子
はい、
について。
そうですね、2点目に関しては、この数字の内訳までは、このデータからは見えないんですけれども、当然、後発品は含まれておりますが、後発品がこの金額換算での市場の伸びを牽引するとは考えづらいですので、やはりここは一定新薬が、市場を牽引していると読み取るのが正しいのかなというように思っております。
ありがとうございます。
はい、ありがとうございました。柳本さん、本当ありがとうございます。体をご自身、ちょっと患者さんのお話してますけれども、体調悪い中ですね、本当プレゼンしていただいて本当にありがとうございました。ご自愛、くれぐれもご自愛いただければと思います。
どうもありがとうござい
柳本
ました。
さんでした。どうもありがとうございました。
個別にご連絡ください。失礼します。
はい。では、それではですね、引き続いて、次はですね、ビットバイオーム株式会社創業者取締役 CSO、そして早稲田大学の准教授でいらっしゃる細川先生からですね、マイクロバイアルジーンビッグデータ for バイオマニュファクチャリング というテーマでですね、またプレゼンテーションをしていただきます。細川さん、どうぞよろしくお願い申し上げます。
はい、よろしくお願いいたします。早稲田大学とビットバイオムの細川と申します。今日こちらのリンク J の前に立たせていただきました。こちらに来させていただくのが、2回目になっておりまして、4年前に、ここでやらせていただきました。私、今日また対面でこのような場を機会いただきまして、大変ありがたく思っております。私どもはビッグデータ、特に微生物の遺伝子のデータを扱っていまして、合成生物学と合わせてバイオものづくりの分野での、事業支援みたいなことをサービスとして行っています。で、僭越ではありますが、バイオものづくりというものはどういうものかというところからですね、ちょっとご説明をさせていただきたいなと思っております。で、今日は UCSDA とのコラボということで、まずアメリカの動きからなんですけども、アメリカではですね、昨年の9月にバイデン大統領から、バイオテクノロジーとバイオマニュファクチャリングに関するイニシアチブというのが発令されております。そこでは、プラスチック、燃料、そして医薬品などの分野におきまして、新たな持続可能な代替製品を提供するポテンシャルが、このバイオものづくり、バイオマニュファクチャリングにはあるというふうに言われています。で、ここに戦略的な投資を行っていくということが宣言されているわけなんですけども、その理由は、このバイオものづくり分野が非常に伸びていくということが期待されているからでありまして、今後10年以内に、あらゆる製造における3分の1が全てバイオに置き換わるだろうと。で、そこでは、日本円で言いますと、430兆円ほどの規模になるということが言われています。で、これが政府から、アメリカのホワイトハウスが出ているわけなんですけども、実際民間の投資というのも盛んになっておりまして、アメリカでは21年から22年の間に、日本円で言うと 4.6 兆円もの投資が行われております。で、お隣、中国におきましても、2年間で890億円。日本は民間投資のデータが残念ながら私の手元にはなかったんですけども、非常にこのバイオものづくりの方に戦略的な投資を行うということで、政府、ネドの方でバイオものづくりに関するさまざまな基金が立ち上がっておりまして、今後10年以内に総計で4800億円もの投資がなされています。そして2030年には、官民合わせての年間投資が3兆円を目指しているので、アメリカ規模に日本が追いつくぞというものが戦略的に考えられているということになります。で、このようなバイオものづくりにおいて、重要なところはですね、バイオテックというと、やはり創薬というヘルスケアの部分が連想されるわけなんですけども、それ以外の化学、素材、食品、そして燃料、こういったおよそこれまでバイオと結びつかなかったようなものですら、今後は化学、石油で作ってみたものをこのバイオベースの作り方に変えるとか、そういったことでバイオに置き換わっていくからということになります。つまり、バイオものづくりというのは、医薬品の製造を指している言葉ではなくて、こうしたいろいろなあらゆる産業におけるものづくりをバイオに置き換えていくものだというふうにご認識いただきたいと思っています。で、アメリカの方でですね、合成生物学の世界的なイベント、新バイオベータというものがあります。私もこちらに出展参加してきました。でそこで、実際に注目されているアプリケーションの事例というものは、もうすでに医薬品以外に転じてもう実用化されつつあります。例えば、カーボンネガティブ、石油化学的には合成をしていた、化学工業の基幹部品を微生物由来の酵素や無細胞翻訳システムを使って、こういう工場の中で作るということが行われております。また、生物由来のタンパク質を使って、衣類の繊維を作るとか、藻類のタンパク質を使いまして、それを代替樹脂としてスキーの板とか衣服を作るとか、 キノコの菌糸を使って車のシートのレザーを代替部品で作るとか、そういったものがだんだん出てきてるんですね。こういうものを作っていく、バイオの力で作っていくというものがバイオものづくりです。そしてここで重要なのが、微生物の持っている力が活用されているということでありまして、我々の身の回りにはものすごく膨大な微生物がいます。私たち自身の体の中にもいて、腸内細菌などを含めると、私たちの人間の細胞の10倍もの細菌がそこに住み着いていますし、土壌の中にはスプーン一杯の土の中にも地球の総人口を超えるほどの微生物が存在しています。こういうものをですね、私たちは古来より醸造とか発酵とか医薬品製造などに目に見えないものを使ってきたわけです。今後のバイオものづくりがどうなっていくかというと、この目に見えないものからもうちょっとデジタルの情報とか AI とかロボットを使ってものづくりをしていこうということになります。微生物からゲノムのシーケンスを読んで、それらをコンピューターの中で設計して、その遺伝子の回路とか改変をデザインし、それから酵素とかスマートセルというのを作って、これまで、生き物、自然界にはなかったようなより良いものを作るとか、そういったものでいろんな素材、燃料、食品、いろいろなものを作っていこうというのがバイオものづくりです。で、このバイオものづくりにおいて重要な研究開発要素というものがよく言われておりまして、これが DBTL サイクルと言われるんですけども、まずその遺伝子の設計図とか、そういったものをデザインして、そこから実際に組み立ててみる。その遺伝子の細胞の中に組み込むとか、そういうことですね。そして出来上がったものを、実際に機能するかどうかというのをテストする。でテストした結果をまた学習して、次のデザインに生かしていく。こういったサイクルを回していくということが重要になります。でこのサイクルをいかに早く回すか、DBTL の期間を短縮するということが非常に重要だというふうに言われているんですけども、この短縮の部分で重要な要素と言われるのが、バイオファウンドリーという、その DBT の設計開発を非常に大きな規模でやるという、設計工場と、もう一つ我々が大事だと考えているのが、その基幹部品となる遺伝子、微生物の情報ですね。情報をたくさん持っているというものは、こういうバイオものづくりにおける、開発候補品、設計図のようなものをたくさん携えているということになりますので、将来長くにわたって競争力を保有するには、こういった誰も持っていない秘伝のレシピみたいなものを持っておくということが大きな強みになります。これを使ってうまく活用していけば、化学とか食品とか燃料、そしてもちろん医薬品も含めて、いろんな部分で、活用が見込めるのではないかと考えているわけです。日本はじゃあこの微生物を利用するにあたってどうかというふうに考えますと、日本の国土というのは地球上の総面積で考えると 0.25% しかありません。ですが、日本というのは非常に多種多様な生物が存在しておりまして、微生物も同様です。これまで見つかった菌類、世界中で見つかった菌類の 15% は実は日本で取れるというふうに言われています。0.25% から考えると非常に大きいと。なぜかというと、日本は四季があって、亜寒帯から熱帯まで、いろんな気候があって、しかも離島が多いということがありますので、いろんな微生物がそこに住み着いているということがあります。じゃあこれ非常に資源国じゃないかというふうに考えられるんですけども、微生物を扱うにあたって非常に大きな問題点があります。微生物、地球上には10億種、いやむしろそれ以上存在するという推定もあります。ですが今見つかって名前がついている微生物は2万種しかおりません。つまり、パーセントで言うと 0.0001% 以下だと見積もれます。なぜかというと、この微生物、これまでは、取ってきて、研究室の中で分離して培養して増やして調べなければいけなかったと。非常に手間がかかる。場所も取る。 労力もかかるし、良いものが取れるとは限らないという、そういった作業だったわけです。ですが、ここでですね、DNA のシーケンス技術の発展というものの手助けになります。その微生物そのものを育てることができなくても、管理することが面倒でも、DNA を一度読んでしまえば、それは永久に残るデジタルデータとして保管することができます。今後、バイオものづくりを行おうと思ったときに DNA の塩基配列さえあれば、そこから必要なものを設計図として組み入れて作り出すことができます。このような微生物の DNA データを膨大に集めてバイオものづくりに生かそうということを我々ビットバイオムという会社で考えています。私たちが掲げているのは Unlock the Full Potential of Microbes ということで、地球に生息するあらゆる微生物の可能性を我々の技術で解き放っていこうという考えです。私たちは2018年に創業いたしまして、もうすぐ5年を迎えます。これまで約10億円ぐらいを調達しておりまして、日米でこちらに示すようなですね、アワードもいただいております。場所は東京、新宿、早稲田大学の中にラボとオフィスを構えておりまして、活動を続けております。また最近はですね、米国でも事業開発拠点というのを設けておりまして、海外の合成生物学会社、あるいはいろんな産業、会社との連携を強く進めております。で、どういう会社かと申しますと、3つ強みとなる技術がございます。1つ目は、微生物の遺伝子を大量高効率に収集することができる微生物ゲノム解析プラットフォーム、ビットマップと呼んでいるものです。そしてこのビットマップを使いまして、積み上げて自社に保有している遺伝子のデータベース。これ、民間企業としては、世界最大級の微生物遺伝子データを今保有しているんですけども、そういうものを、ジーンエンサイクロペディア・オブ・マイクロブスというふうに名付けて、頭文字取ってビットジェムというふうに呼んでいます。そしてこの遺伝子のデータベースの中には今いろんな産業利用できる酵素の遺伝子もたくさん含まれております。これらをですね、AI の技術を使って構造を予測する、そして機能を予測するということや、またロボティクスを使って実際に作ってみて、どんな機能を持っているのか評価するというようなこともできまして、こういうものを使って品質の良い酵素を見つけていくという、クオリティエンザイムディスカバリーということで、ビット QED という技術を保有しております。つまり、遺伝子を収集し、それらをそこから宝物を見つけ、宝石を磨き上げていくというような、この3つの技術を持っているわけです。一つ一つについて簡単にご説明します。一つ目はビットマップです。ビットマップはですね、早稲田大学の方で開発した技術で5つのステップからなっております。まずはじめにマイクロ流路と呼ばれるもので、微生物の含まれるサンプルを小さなカプセルの中に封じ込めていきます。ここで細胞一個一個に分離されていきます。このカプセルは特殊な構造をとっていて、その中で DNA を抽出して個別に増やすということができます。ここで1細胞別に付属されたこのカプセルを回収してまいりまして、細胞1個ごとにゲノムのシーケンスを行っていきます。このようにすることで、どんなに複雑なサンプルであっても細胞1個ごとに微生物データを収集することができるので、このデータベースの中で欲しい遺伝子があれば、それを文字列として入力すれば検索することができるというものです。このような方法を用いて、例えば土壌の微生物のゲノム解析を行うと、従来の方法だと1万個の遺伝子が見つかるのに対して、我々の方法を使うと30万を超えるような遺伝子をいっぺんに収集することができます。一個一個細胞のゲノムを調べることによって、一見めんどくさそうに思えますけども、非常に高効率に遺伝子を収集することができるというものです。でこのようにして積み上げた遺伝子の総数は12億に到達しておりまして、1年あたり10億遺伝子、さらに蓄積を追加できるぐらいのスピード感です。 で、こちらで示している水色のところは公共のデータベース。世界中の研究者が論文を書くときにこういうデータを登録するわけなんですけども、そこに比べても公共のデータベース、一番大きいものにももう数年以内には追いつき追い越す状況です。そしてこの遺伝子たちがどこからやってきたかというと、日本の環境の微生物なんですね。土壌とか、海洋とか温泉とか、鉱山から実際我々がサンプリングして取ってきた未知の微生物の遺伝子です。で、これの集積スピードをどういうふうに捉えればいいかというと、従来の技術を使うと世界中、地球上のサンプル、微生物数をすべてゲノム解析しようとすると、1万年かかって、非現実的な時間がかかっていたというのに対して、我々の技術を使うと、100年以内、ひょっとしたら自分が生きている間に、すべての微生物データを収集できるぐらいのスピード感だということになります。そしてこの微生物遺伝子データ、集めるだけではありません。使えるように磨き込むための技術も持っています。まず、エンザイムをマイニングする。遺伝子のデータの中から目的の酵素の配列データを、検索ボックスに入れるようなイメージで探します。で、配列が似てるだけではなくて、酵素というのは形でその機能とか安定性が決まりますので、3次元の構造予測技術、アルファフォールド2なんかを使いながら、こういうものを、酵素をですね、インシリコでスクリーニングします。で、そこで出来上がったものをさらに実験的に候補を絞り込み、候補品のショートリストを作るわけですね。で、そこでできたものをさらに、機械学習の技術とかロボティクスを使って磨き上げて、さらに性能を良くする。例えば周率を10倍に上げる。高温で作用する酵素を獲得する。そんなことができるわけです。これらは従来狙ったものを取るということは難しかったんですけども、私たちの方法を使えばいろんな遺伝子のプールの中からそれらを見つけ磨き上げる。これを数ヶ月で実行することができます。一つ事例をご紹介したいと思います。プラスチックの廃棄、そして資源循環の問題、大きな課題になっています。で、このプラスチックが世界に溢れるようになって、プラスチックを分解できる微生物というものが現れてきました。で、近年こういった研究進んでいるんですけども、プラスチック、例えばペットボトルのペットを分解できる微生物、いくつか見つかっているんですが、そういうものはどこから見つかってくるかというと、ペットボトルが廃棄されていそうな土とか海とかコンポスト、堆肥を作るためのコンポストの中とか、あるいはそのプラスチックを食べることができる虫の腸内細菌なんかを持っています。で、こういう微生物のゲノムデータを僕らは調べることができるので、実際にやっていました。社員の家の脇の土を実際に取って、あとはコンポストから土を取ってやってみました。そうすると本当に見つかるんですね。プラスチック分解酵素が。で、1個事例をお示ししますけども、このプラスチックのペットのフィルムが分解できるんです。これペットのフィルムですけども、酵素の液にただ入れて2週間そのままにしておくだけで、だんだん白くなって表面がざらついてきます。2週間ちょっと経つともう、ここからここが劇的なんですけども、完全に消失します。で、消失するんですけども、これ最終的には小さな部品に変わっていますので、そこから将来的には再合成できるんじゃないかなというふうに考えているんですけども、こういうの見つかっただけではなくて、改良することによって、競合品相当の効果を得ることもできます。で、こういった酵素を探して育てる、データからですね、ビッグデータから実現することができますので、今、NEDO のプロジェクト、GI 機器のプロジェクトでは、CO2 を固定してカーボンリサイクルによってものづくりをするということで、製品物質評価基盤機構さんらと協力して、日本のいろんなところから微生物をサンプリングしてきて、そういった微生物の株や遺伝子のデータを今後のものづくりに役立てるためにデータベース化するというような仕事を今年度から始めました。で、また、医薬品の部分でもですね、科研製薬様と感染症薬の創生に関わる共同研究もスタートいたしまして、環境問題からこういった医薬品開発につながるところまで幅広く我々の技術を応用していきたいなと思っています。 このように私たちの会社自体はね、本当にゲノムのデータの分析から始まっているんですけども、そこからコースを探して改良するということも始めまして、今年からもう本当に単品コースを製品として作るということも始めています。将来的にはもっと複雑な部品をいろいろ設計して、バイオのプロセスを革新していくということもやろうと思っています。で本当に今、このバイオものづくりということはいろんな部分で使われますので、バイオに馴染みのなかった方でもですね、こんな分子をバイオでやりたいんだけどみたいな相談をもう実際に受けて、そこの、ヒアリングをして一緒に考えるというところから実はやっておりますので、ぜひいろいろご相談いただきたいと思いますし、これまで実現できなかったものづくりをですね、このバイオの力で、そして日本初、日本の資源を使って実現したいと思いますので、ぜひお声かけください。本日は今日もありがとうございました。
細川さん、ありがとうございました。非常にスコープの大きいバイオマニファクチャリングとはというお話から入ってですね、ビットバイオム社の最先端の取り組みまで非常に興味深いプレゼンテーションだったかと思います。ご質問の方いかがでしょうか。お時間ありますが、の方はあんまないんですけれども、1問ぐらい、もしよろしかったら、お願いします。
楽しいお話ありがとうございました。今お話聞きながらこれ AI と比べてみたんですね。いろいろ細菌とかですね、カビも含まれるだろうと思うんですけども、あちこちから取ってくると。で、ラベリングはしないかどうかという問題なんですね。要するに、持ってきたもの、一体それ何なの？名前をつけないといけないですね。で、それの大体の性質を見る。で、最終的にはその遺伝子解析でどういう構造がどういう機能を持つかというところまで持っていくんだろうと思うんですけども、非常にいっぱいものがあって、なおかつ突然変異がしょっちゅう起こってるわけですよね。だから最終的な目的は分かるんですけども、それに至るまでっていうのは相当複雑なパターンになってくると思うんですね。で、その時にじゃあある程度分かったらですね、これジェネラティブ AI を使って逆にものを作っていくと。それでデータを消すとかですね。なんかそういうことが考えられるのかなというふうにちょっと聞きながら思ってたんですけど、その辺いかがでございましょうか。
ありがとうございます。実際にですね、タンパク質の3次元構造を予測するアルファフォールドとか、Google の傘下のディープマインド社が作っているものですけども、そういうものも実際に何を学習させているかというと、世界中で捉えた遺伝子データをもとに学習させて、そういう予測ができるようになっています。で、我々みたいなこのデータがまた積み重なっていくと、おっしゃられたようなジェネラティブ AI みたいなもので、海外ではですね、実際抗体とかをこういうデザインにしてやりたいんだっていうのを入れて、ジェネラティブ AI で作るみたいな企業も実際出てきておりまして、おそらくこの数年内にそういう動きになっていくと思います。で、そういう企業からもですね、お宅のデータ使いたいんだけどってやっぱ来るんですよね。ですからそういうデータを保有していく強みっていうのはこれから出てくるんじゃないかなと我々は思っております。
はい、ありがとうございました。貴重なお話、本当にありがとうございました。お時間の関係で、じゃあ、ここまでとさせていただきます。それでは、もう一度、細川さんに、大きな拍手をよろしくお願い申し上げます。それではですね、全体の時間若干押しておりますが、これからセッション3の方にですね、引き続き移らせていただきます。こちらはですね、パネルディスカッションでございます。バイオマニュファクチャリングの将来ですね、こちらに関するディスカッションとなります。ご登壇いただくのはですね、第1セッションでプレゼンテーションをしていただきました、バーカート先生、そして相沢先生、そして、まさしく今ご登壇いただきましたセッション2のですね、山口先生にご登壇をいただきまして、モデレーターはですね、ピサの学部長、に、お願いします。はい、それではよろしくお願いいたします。
Okay, thank you. Thank you very much. And I want to start by thanking all the speakers for their wonderful presentations. We really appreciate that you can share valuable insights with us. Okay, so I'd like to get started by asking a question, but first I'll offer something that I thought I heard from each person's presentation. Each person in their presentation pointed out at least one step that was causing them difficulty. One place in their process where they either needed more speed, more data, more resources. Okay. So with the idea that we're all working as hard as we can, but if we're going to win this innovation race, we have to figure out how to go faster. I'll ask each of the speakers to begin by saying, what is at least one thing? You don't have to say everything, but name at least one thing that would help you go faster in the future. All right, so Michael, I'm going to start with you. Switch.
So I would say there are wonderful computational resources available, but they're all separate from each other, right? And to have the ability to link these together, ideally with some type of AI package, would really speed the processes that use computational tools.
Okay, Michael, thank you very, very much. Our next panelist.
Me? Okay.
Yeah, straight in a
Okay,
row.
I would say, okay. It's a difficult question, but because I'm kind of, like I said, I'm managing this small startup right now. So I think that for innovation, the most important resources, I should say, is the people, talented people. So yeah, especially in the bio production in Japan, we've seen not so many people working on the field yet. I think so that we have to kind of rush to make some environment for the education and also education of young people, as well as re-education of the grown-ups from different sectors. That's very important, I think.
Okay, thank you. Maybe your comments.
Yeah, basically, as you know, I'm living in the pharmaceutical, sector, which is a highly regulated area. So therefore, I would say, yeah, most important, yes, we can leverage the emerging technologies like AI and robotics, etc., as I presented. However, in such a situation, most important thing is, to develop the new regulatory framework, because we are regulated, we are controlled by regulatory agency, which you can do, like child. So therefore, in combination with in taking new technology and to newly developing the new regulatory framework is, I guess the key for especially pharmaceutical industry.
Okay, thank you, thank you. So in one of the speakers' presentations, I remember seeing a comparison of research investments in the field, and I think it was our second speaker, not yours. And I was very pleased when the speaker said, Japan is on track to be making similar levels of investments of the United States. So I took this as a signal that the two countries are ready to be partners. Each one is investing a similar amount, and therefore there is automatic parity, and that means that collaboration is much easier to achieve. Okay, so let me rephrase a question now using that as a basis. If it is true that we're seeing the governments and the investors of each country starting to make similar investments, similar sized, then if these investments were to be made for infrastructure that could be commonly shared to the benefit of all, what kind of infrastructure do you think would be useful? And again, one after the other, Mike. I think you were mentioning something already in that direction.
Sure. So, I mean, the computational connectivity is going to be really important, and being able to have groups work together. Again, bringing AI into this, having groups work together and participating with their expertise, I think is going to be really important. We have big data. it sits somewhere, but you have to be able to do something with it. And then you need an output that has value. And those aren't necessarily joined together right now.
Okay, thank you. So our next speaker had mentioned people shortage. I'm just curious to see if you would explore that in the context of what kind of research infrastructure investments could be made to help relieve your problem.
Yeah, that's what I'm thinking actually. So yeah, as you know, these days the areas of each academic researchers are working on, it's very tiny, it's very narrow. But I think for the pushing this kind of field, we have to have some interconnections of the different disciplines, right? Definitely. So yeah, I think yeah, and in terms of making a good human resources and also the good human connections, we have to have some infrastructure to merge the kind of different talents from different disciplines for sure. And actually, just probably some of you guys noticed that these are education ministry now open some actually the grant right now to make infrastructure of this kind of bioproduction right now. Actually, the deadline is very soon. Yeah, so we're moving in the direction right now.
Excellent, excellent. Our third speaker, do you think you have any a suggestion as well?
Yeah, I also would like to touch upon the human resource, so especially for biomanufacturing, area. Also, Japanese company are also struggling in securing the enough amount of operator. So therefore, human resource issue not just for the PhD science people, because the problem is, government try to create so many PhD scientists, but actually those in scientists don't want to work at the manufacturing site at operator. So therefore, some kind of operator level, human resource lack is also a problem. So therefore, that kind of government investment would be good to be spent for such an aspect as well. That's my point.
Okay, thank you very, very much. All right, so now I'd like to look at the audience, both remote, online, and here in the room. You've seen me push on each of the speakers for a set of answers. Is there anyone who wants to ask a question, any question, English or Japanese, to any one of the speakers? Can we get a question from the audience? There's one online.
There is one online to Dr. Burkhardt. Thank you for your presentation. How do you find the amino acid to be mutated using AI or by your idea?
Sure. So what I presented hasn't yet been published, although it's a paper, it's in review right now. We use a protocol based off of Rosetta, which is a computational framework that was developed by David Baker at University of Washington. And Rosetta has multiple suites, but what we do is we begin by using a docking protocol that takes the two proteins that initially have no affiliation with each other, and we dock them together based on a pose that comes from one of those crystal structures that we had collected from the enzyme. And then we have Rosetta look at the surface of the two proteins and propose mutations at different positions in silico. This is done inside the computer. And then it performs the docking once again to see if they bind together better. And that process repeats 1,000 times. So by using that computational framework, the program can then recommend certain sites to do the mutagenesis. We go into the laboratory, we mutate those sites, and we test it enzymatically to see whether indeed they match or not. And that data goes back into the computer, and it goes through another round of selection. So it's really in vitro evolution using computational methodology. And as I was saying before, this really could be accelerated by being able to draw from different types of software, different types of structural databases, different types of genome databases that exist, to make this a much more kind of robust pathway.
Okay, thank you. Thank you, Mike. Are there any other questions from the audience or from online? No? Okay. Yes, great. One moment, the microphone comes.
すみません。日本語で失礼いたします。マイケル先生にちょっとお伺いしたいんですけど、代謝の制御の大腸菌について、質問がありまして、その代謝を制御した大腸菌のデータは、大学にあったりするんですか？自分たちで全て作ったのか、それともその元になるものがあって工夫されたのか、どちらかちょっと教えていただけるとありがたいと思います。
Let me see if I can make sure I understood your question. So the ability to do this type of engineering approach, is that technology that's open? Is that correct, whether it's been patented or not? Sure. So this is really a technology that we don't have IP on because it's based off of methods that are already available, right? So this Rosetta technology is available to anyone, and so people can utilize it, and we're really just making use of data that we collected, crystal structures that we collected, but those are deposited to the PDB database. So this is really more of a methodology that's available to anyone. But it's still in its infancy. We haven't yet engineered organisms for production. I hope that answered your question.
ありがとうございます。
Yes, we have another question here.
I'd like to answer for all of you, but maybe Michael might be the best person to answer to this question. With respect to the database connection, you mentioned the different database assets and something so forth. With respect to do the biomanufacturing, it is very difficult to have the standardized database. There's a global database there, but it's not necessary to have all of the factors in the same sets with the sub-factors to be input. And with respect to the partnership or the collaboration to be made between US and Japan or between the companies, how do we have the standardized database to be established internationally, or at least between the US and Japan?
Well, I start with a caveat. I am not a computational chemist or scientist. I'm a user of those databases and those technologies. What was beautiful about Rosetta is that it was basically made open, and anyone can learn how to use it pretty easily. But you come to a good point, is that when you've got all these different mom and pop databases and each one is independent of each other, how do we work together? And so maybe creating standardizations, if we can between the US and Japan, and work together to come up with these types of standardized models, that would be ideal. But I wouldn't know how to make it happen. I'm sure if Bernard were here, I'm sure that he could have answered that question better.
Yeah. But I think if you think about the competitor, you want to have some standardized data for the competitors, right? So, and also I think what kind of data set we have is the most, how can I say, it's like the selling point to some extent, right? So what kind of parameters we have to control and monitor the system. I think so that could be, yeah. So that's the treasure. So that people cannot share with. That's my answer. Yeah. Sorry.
And Yamaguchi-san, you would agree?
Yeah, basically agree. Yeah, it's difficult to share the, very, very detailed, manufacturing related data with other vendor. And also as just a user of the data, obviously, corporate-wise, we are struggling in handling the each vendor's proposal, totally different format by format. How we can consolidate it to the one data, for instance, security, to cope with security issue or to generate additional value, etc. So therefore, I do agree with your point. And as a customer, I'm pretty much, hope to have some kind of consensus among the IT company or government. Yeah.
Great, thank you. Please continue.
Yeah. Yeah. I mean, I think I'm kind of taking the opposite side from what I said. But on the other hand, probably I think the open innovation concept should be more, like I said, here in this field as well. So I think as most of you guys know, I think the concept of open innovation is the core of the open innovation must be how we can design the boundary between the open part and the closed part. And then I think, yeah, we have to have some effort to design the open part for the synthetic biology, I think. I mean, that will accelerate everybody's, I think, the development for sure. Thank you.
Okay, Aizawa Sensei, thank you, thank you. I understand we have another
Thank
question.
you very much.
I apologize.
No, it's okay. But thank you very much for.
Thank you. Please forgive me. I was getting a signal that we have another question online, and I wanted to make sure we hear that.
Yes. So thank you so much for valuable opportunity. I was wondering if I could ask about data integration. Combining large amounts of data from different sources can be challenging. This can make it difficult to draw meaningful conclusions from the data. How do you think about this issue? Also, there is a data privacy as well.
No particular target?
So this, I read the question.
Go ahead. All right, panel, does anyone want to tackle that one?
I think we kind of, these were the topics that we were just discussing, but if we want to think about ways to handle the data flow actually being communicated properly. Again, I think that we can turn to artificial intelligence in order to make databases match up with each other. That is the power of this kind of technology, but we need people doing that kind of work. And I don't think that's any different from the message that I was talking about earlier. In terms of data privacy, yeah, that's going to really, I think, depend more upon who generates the data and whether, if we generate data that's funded by, say, the NIH in the United States, we have to make that publicly available. And really, a lot is going to depend upon the funding source. And, for example, if it's patient data, by definition, there has to be privacy behind it.
Any other answers forthcoming? You look tempted. No? Yes? Okay, go ahead.
So just one point. So in terms of, I think, economic security stuff, these days, it's one of the hot topics in the technology transfer, right, between the countries. So in that sense, yeah, we have to have some regulation to protect IP for sure. So yeah, I think the government's working on that. I hope so. Yeah. that's not our territory. That's not our territory probably yet.
Okay, thank you. We have time for one more question. Just 5 minutes to go. One more question. Is there anything online? ChatGPT has provided no questions. Oh, okay. All right. And last chance, anyone from the audience? Last chance, anyone from the audience? Any other questions? No one's asking what's being served at the reception. Not even that question. Oh, yes, a question. One moment. Here comes the mic from behind you, here in the front.
My question might be a little bit different from the different scope as you are looking at. But I'm very much interested how you will accelerate the collaboration and research for your target. And so far, look at the history of industry in the world. All are target based on mathematics. And as I say, consistency of the idea through mathematics works for engineering and physics and of course medicines and physiologies. So therefore, how do you think the mathematician should be integrated in new research or project to accelerate your, to get target?
Okay, our panelists, is anyone interested in trying that one?
Thinking seriously. Okay. So, well, I think, yeah, thank you very much. Very tough question, but mathematician. So, but I should say, I think the communication even between the biologists are very hard right now. And then, as I mentioned earlier, I think that these days, each researcher working on the very narrow discipline right now, so that the communication between the researchers in the different disciplines is still difficult, actually. But I think your comment must be like, maybe if we simplify our demand, like in mathematic formula, maybe we can accelerate the communication as well. And then probably the issues we have in our mind, probably not sophisticated yet, probably. But if we narrowing down and digest the issues we are struggling with every day into the very simple some formula, probably we can simplify, maybe categorize the issues most in a simple way, and then maybe that will accelerate the communication between the different researchers in the different disciplines, probably. Yeah. So, yeah, that's my answer. Yeah.
Okay, Yamaguchi-san, you were thinking of an answer?
Yeah, I think obviously, mathematics is, every, how can I say, academic basis. So therefore, there's no argument, the importance of the mathematics. And for my area, pharmaceutical area, we have a tons of molecular biologists and synthesis chemists. But now, progress of the emerging technology like robotics, AI. So, what kind of we are struggling in the, not all, PhD scientists cannot digest the, new area, especially mathematics related area. So, therefore, kind of, mathematician who can give us the, how can I say, digest the, contents of research content, to us easily or understandable. That is quite beneficial. So therefore, for my area, we are intentionally trying to hire the, not only molecular biology, but also chemical engineer or physics, who got major physics, etc., etc. So therefore, trend is now getting little changed, because of the emerging of the new technology like AI. So that kind of situation. So therefore, yeah, not a direct answer to your question, but I would say, yeah, mathematics is quite important for bioindustry as well. That humble basic conclusion. Sorry.
Okay, so we are at time. I want to thank all. Oh, we are at time. I want to thank all our speakers. Thank you very, very much, panelists. Wonderful job. Thank you very much to the audience, both here and online. Everyone was very eager to ask a question, and I was pleased to hear all the questions come forward. Thank you very much, and I think the panel session is now closed.
「クロージング」ということで、日本語でさせていただきたいと思います。皆様、本日はお忙しい中、ご参加いただきましてありがとうございます。このシンポジウムを企画しましたのは、もちろん昨年バイデン大統領の今日のお話でもありましたけれども、バイオマニファクチャリングやるぞというアメリカの意気込みが示されまして、それに対して日本はどうなっているのか知りたいなっていうのが一つと、今日のお話の中でもずっとじゃあコラボレーションっていうのでどういうふうに何ができるのかっていうのをちょっと掘り下げてみたかったので、こういう機会を設けました。ぜひこれに今回で終わりにせず、また次回も何かちょっと違った形でこういう機会を持てれば双方にとっていいんじゃないかなというふうに思ってます。UC さんではいろんなことできますので、ご質問ありましたらぜひ私なりまた参加者のものにお問い合わせいただければと思います。ありがとうございました。じゃあちょっと津山さんの方に最後のコメントをいただきたいと思います。
はい、和賀さんありがとうございました。和賀さんはですね、長年 UC サンディエゴ校でオフィス・オブ・リサーチ・アフェアーズ、その中でも研究成果を海外も含めた学外に出して、実用化につなげていくアウトリーチを担当なさっている、シニアディレクターで、いらっしゃいまして、Link-J Ucさん冒頭ご挨拶申し上げたようにですね、設立前から大変、助けていただいている方でございます。今日はですね、バイオものづくりということで、多くのご登壇の皆様からさまざまな切り口で、そして最先端の取り組みもご紹介いただきましたし、最後のパネルではですね、いろいろと深いお話、ディスカッションも、させていただきました。で、途中、本当はパルソン先生の、ビデオをご紹介いただくする予定だったんですが、ちょっとすいません、準備の、不出来でですね、音の方が、ちょっと悪かったので飛ばさせていただきました。こちらに関してはですね、アーカイブの方、今回の、シンポジウム、アーカイブしようと思ってますので、その際にはですね、音もクリアになったものをですね、アーカイブいたしますので、ぜひそちらをご覧いただければなと思います。最後ですけれども、ディスカッションでもございました、やはりコラボレーション、これすごく大事だなと思います。この後ですね、会場の皆様はですね、ネットワーキングの時間もご用意してありますので、ぜひですね、そちらで、ご登壇の皆様、またはご参加の皆様同士でですね、コラボレーションのためのきっかけ作り、どんどんやっていただければなと思います。UC サンディエゴ校はですね、先ほど和賀さんおっしゃっていましたようにですね、バイオ、ライフサイエンスだけではありません。私、前回行った時はですね、レセプション出たらちょうど他のレセプションに、同じ会場でやってて、宇宙飛行士の方が2人ぐらい来ていたということで、航空宇宙もはじめですね、さまざまな分野で最先端の、取り組みなさってますので、ぜひですね、UC サンディエゴ校とのコラボレーションもですね、進めていただければと思います。それではご視聴の皆様、ご参加の皆様も含めて、和賀さんがまた一言。
すみません、最後にもう一言、冒頭、すみません、須山さんの話の中でもありましたけれども、UC サンディエゴは、ビヨンドジャパンというプログラムのアクセラレーターに選ばれまして、日本初のライフサイエンスの、企業、ライフサイエンス系スタートアップの海外展開を助ける仕事をすることになりました。で、これまでもたくさんの大学系のスタートアップさんの研修というかアントレプレナーシップトレーニングやってきたんですけれども、本当に私どもの持つネットワークを生かしてなるべく成果が出るような形のトレーニングをしたいと思ってますので、ぜひ今回ご参加いただいている皆様およびオンラインでご視聴いただいた皆様、ぜひ募集要項の方を見ていただいて、ぜひぜひ応募いただければと思います。皆様とサンディエゴでお目にかかることができるよう祈っておりますので、よろしくお願いいたします。
ありがとうございます。ビヨンドジャパンとウェブで引いていただいて、いくつか出てくるんですけれども、その中でジェトロさんがこれやられてますので、ジェトロというのが入ってる、ところをご覧いただくと、公式なホームページも立ち上がっておりまして、詳細出てますので、ぜひそちらをご覧いただければと思います。それでは今日はですね、オンラインでご視聴の方、そしてリアルでですね、ご参加のいただいた方々、もちろんでございますが、ご登壇をいただいた皆様、本当にありがとうございました。これにてですね、ネットワーキングより前のセッションの方はこれにて終了させていただきます。ありがとうございました。